Welcome. Tonight we are exploring the genetic mystery of hazel eyes, a rare trait requiring the orchestration of dozens of different genes and an optical illusion created by the physics of scattered light. This is the story of how an ancient genetic mutation traveled across continents, intertwining deep human history with the microscopic mechanics of the human body. If you enjoy drifting into these slow, factual journeys, please like this video, subscribe to the channel, or leave a quiet thought below. Settle into your space now. Soften your shoulders. Let your breathing slow and lengthen. Let's begin. The time is 300,000 years ago, during the Chibanian stage of the Middle Pleistocene epoch. The location is Jebel Irhoud, a limestone massif located 50 kilometers southeast of the city of Safi in modern Morocco. The geological formation consists of sedimentary rock containing heavy deposits of barite and iron ore. The surrounding landscape features a dry, open savanna ecosystem populated by drought-resistant shrubs, wild olive trees, and perennial grasses. The daytime temperature peaks at 38 degrees Celsius, heating the red clay and fragmented limestone pebbles underfoot. Early Homo sapiens inhabit a network of karst caves eroded directly into the hillside. The skeletal structures of these individuals exhibit physical features matching modern human anatomy, including a flat facial profile and reduced brow ridges. Their skulls contain a brain volume averaging 1,400 cubic centimeters. An adult male measures approximately 170 centimeters in height and possesses heavily muscled limbs adapted for endurance, walking across the rugged terrain. Outside the cave entrance, an adult human shapes a core of flint using the Levallois stone knapping technique. The human prepares the stone by chipping the edges to create a convex tortoise-shell shape. The individual strikes the prepared edge of the flint core with a rounded quartzite hammerstone, applying precise kinetic force to detach thin, sharp flakes of stone. The friction between the rocks generates a smell of burning sulfur in the dry air. The human selects a triangular flake measuring 7 centimeters in length to process an animal carcass. Nearby, members of the group butcher the remains of a hunted Thompson's gazelle. The humans use the freshly struck flint blades to slice through the dense muscle fibers and tendons attached to the gazelle's femur. The raw meat smells of iron and blood. They place the stripped bones near a central hearth containing layers of compacted wood ash and charred sediment. The humans use heavy cobbles to smash the long bones, exposing the lipid-rich bone marrow inside. They consume the marrow alongside roasted acacia seeds and foraged tubers. The sun sits at an angle of 70 degrees in the sky, emitting a continuous stream of electromagnetic radiation toward the surface of the Earth. This solar radiation travels 93 million miles through space and penetrates the Earth's atmosphere. The radiation includes ultraviolet light, specifically UVA wavelengths, measuring between 315 and 400 nanometers, and UVB wavelengths, measuring between 280 and 315 nanometers. These high-energy photons strike the exposed skin and the open eyes of the humans working in the sunlight. The UVB photons possess sufficient energy to penetrate the cellular membranes and break the chemical bonds within human DNA. The human body actively responds to this ultraviolet light exposure. Specialized cells called melanocytes operate within the stratum basale, the deepest layer of the skin's epidermis. The ultraviolet radiation stimulates the melanocytes to synthesize eumelanin. Eumelanin is a highly dense biological polymer composed of carbon, hydrogen, nitrogen, and oxygen atoms. The melanocytes package the eumelanin polymer into microscopic cellular organelles called melanosomes. The melanosomes travel outward through dendritic appendages and deposit the brown pigment directly over the nuclei of the surrounding keratinocyte skin cells. The chemical structure of the eumelanin absorbs the incoming ultraviolet photons. The pigment physically converts the absorbed electromagnetic energy into microscopic amounts of heat, protecting the DNA inside the cell nucleus from degradation. The identical biological process dictates the structural color of the human eye. Light enters the eye through the transparent cornea and passes through a clear fluid called the aqueous humor. The eye relies on the iris, a circular structure measuring 12 millimeters in diameter, to regulate the diameter of the central pupil. The iris consists of two distinct anatomical layers. The posterior pigment epithelium forms the back layer of the iris. This layer measures exactly 2 cells in thickness and remains permanently filled with a solid, opaque layer of black-brown melanin pigment. The anterior layer of the iris, known as the stroma, consists of loose connective tissue, collagen fibers, and blood vessels. In these early African populations, the stroma contains an extremely high density of active melanocytes. These ocular melanocytes continually manufacture high volumes of brown eumelanin. The pigment accumulates thickly throughout the anterior border layer of the stroma. When ambient sunlight hits the front of the eye, the heavy concentration of brown pigment absorbs the light rays immediately upon contact. This rapid absorption traps the incoming light completely within the tissue. The physical presence of the melanin creates a deep brown visual appearance in the irises of every human in the group. The dark pigment absorbs scattered light inside the eyeball, increasing visual contrast and protecting the delicate photoreceptor cells in the retina from the severe glare of the midday sun. The biological instructions for this heavy melanin production reside on human chromosome 15. The genetic sequence ensures the synchronized production of high melanin levels in both the skin and the eyes. Every Homo sapiens living during the Middle Pleistocene carries this specific genetic sequence. Every individual in the human species possesses deep brown eyes. The genetic code passes unchanged from generation to generation through the natural reproduction of these nomadic hunter-gatherer populations. Around 60,000 years ago, the global climate enters a glacial period, dropping sea levels and opening new land bridges. Small bands of humans begin migrating north out of the African continent toward the Eurasian landmass. The nomadic groups follow migrating herds of grazing animals, including fallow deer and wild boar, across the Sinai Peninsula and into the Levant region. They establish new seasonal camps in the Zagros Mountains and the Anatolian Peninsula. The populations travel into higher latitudes characterized by dense cloud cover, weaker seasonal sunlight, and significantly lower levels of ambient ultraviolet radiation. The bodies of these humans require ambient ultraviolet light to synthesize vitamin D in the skin. The ultraviolet B radiation penetrates the human epidermis and strikes a cholesterol derivative named 7-dehydrocholesterol. The photon energy breaks the carbon bonds within the 7-dehydrocholesterol molecule, converting the chemical structure into pre-vitamin D3. The thermal heat of the human body, operating at 37 degrees Celsius, rapidly isomerizes this pre-vitamin into cholecalciferol. The human bloodstream transports the cholecalciferol directly to the liver and the kidneys. Inside these organs, specialized enzymes metabolize the chemical into a biologically active hormone called calcitriol. Calcitriol travels to the human intestines and initiates the absorption of dietary calcium and phosphorus. The human skeleton relies on these specific minerals to construct rigid, load-bearing bone tissue. 45,000 years ago, human populations expand further north into the European continent during the Würm glaciation period. These nomadic bands walk into higher latitudes, crossing the Alps and settling into the river valleys of the Swabian Jura in modern southern Germany. The axial tilt of the Earth and the dense cloud cover of the glacial climate reduce the available ultraviolet B radiation in this region by over 50% compared to the equatorial savanna. The regional temperature drops to an average of -5 degrees Celsius during the winter months, freezing the topsoil and depositing layers of snow across the ground. The humans inhabit the Mammoth Steppe biome, an ecosystem spanning from modern Spain across the Eurasian plains. The landscape consists of dry, cold grasslands dominated by sagebrush, sedges, and low-growing mosses. To survive the dropping thermal temperatures of the glacial environment, the humans construct layered clothing from the hides of hunted reindeer and steppe bison. They use sharp awls, carved from animal bone, to punch precise holes into the thick leathers. They thread needles made from mammoth ivory, with dried animal sinew, to stitch the hides together into sealed, airtight garments. These thick layers of animal fur cover 90% of the human skin surface, exposing only the face and hands to the open air. The combination of heavy fur garments and the low angle of the northern sun physically blocks the necessary ultraviolet B photons from reaching the human epidermis. The heavily pigmented skin of these early European migrants absorbs the few available ultraviolet photons in the upper layers of the epidermis, long before the light reaches the 7-dehydrocholesterol molecules in the lower cell layers. The bodies of these humans synthesize inadequate amounts of calcitriol. The resulting severe calcium deficiency causes a structural bone disease known as rickets. The load-bearing bones of the legs bend outwards under the physical weight of the human body, and the pelvic bones deform into asymmetrical shapes. The structural pelvic deformation narrows the human birth canal, causing fatal physical complications during childbirth for the females in the population. Spontaneous genetic mutations occur randomly during the cellular DNA replication of these isolated human populations. Specific mutations alter the SLC24A5 gene and the TYR gene. These genes control the chemical efficiency of tyrosinase, the primary enzyme responsible for manufacturing brown eumelanin inside the melanocyte cells. The genetic mutations reduce the chemical processing speed within the cell, dropping the total volume of eumelanin produced by the human body. The melanocytes deposit fewer brown melanosomes into the surrounding skin cells. Individuals carrying these specific genetic variations possess significantly lighter skin pigmentation. The lighter skin allows the weak northern ultraviolet B radiation to penetrate deep into the epidermis and reach the cholesterol derivatives. These lighter-skinned individuals successfully synthesize calcitriol during the short northern summers. They build strong, mineralized bone structures and survive the mechanical stress of childbirth. Through the strict mechanics of natural selection, the genetic codes for reduced melanin spread rapidly and permanently through the European and Asian populations over the subsequent 20,000 years. The genetic sequences controlling melanin production operate systemically across the entire human body. The tyrosinase mutations drop the melanin levels in the skin, the hair follicles, and the anterior stroma of the eyes simultaneously. The ocular melanocytes inside the human iris slow their biological production of the dense brown pigment. The stroma retains its physical architecture of connective tissue and collagen fibers, and holds a significantly smaller volume of eumelanin in the cellular matrix. The reduced pigment concentration fundamentally alters the optical physics of the human eye. Ambient light penetrates deeper into the front layer of the iris tissue. The light waves strike the suspended collagen fibers inside the stroma and reflect back out through the transparent cornea. The remaining moderate amounts of eumelanin mix with a yellow biological pigment called lipofuscin. The combination of the reflected light and the reduced pigment generates new, intermediate eye colors across the northern populations. The irises of these humans display hues of light brown, amber, and deep copper. The glacial ice sheets begin to retreat 12,000 years ago, marking the start of the Holocene epoch. The global temperature rises, and dense deciduous forests of oak and elm replace the cold grasslands of the mammoth steppe. Human bands migrate east and south, tracking the changing ecosystems toward the shores of the Black Sea. The Black Sea forms a massive inland body of water covering 436,000 square kilometers between the land masses of Europe and Asia. The surrounding terrain features temperate broadleaf forests and fertile river deltas fed by the Danube and Dnieper rivers. The humans establish semi-permanent settlements along the coastal plains, foraging for wild grains, catching freshwater fish, and hunting red deer with flint-tipped spears. The regional climate exposes the human bands to distinct seasonal variations in sunlight, further isolating their specific genetic traits. In a single individual living near the northwestern coast of this sea, a new DNA replication error alters the exact sequence of chromosome 15. This specific genetic error creates a profound biological switch that directly halts the primary biological pathway for melanin production in the front layer of the human eye. Between 8,000 and 10,000 years ago, during the Neolithic period, a human child is born in an agricultural settlement near the northwestern coast of the Black Sea. The settlement sits on a flat coastal plain covered with deep alluvial soil. The inhabitants construct rectangular houses using timber frames packed with a thick mixture of wet clay and dried cereal straw. The humans harvest einkorn wheat and barley using curved wooden sickles fitted with sharpened flint blades. They grind the harvested grain on flat basalt stones to produce coarse flour. The adults manage herds of domesticated sheep and goats within wooden enclosures, harvesting the animal wool and spinning the fibers into textiles using fired clay spindle whorls. Inside the cells of this specific child, an exact biological sequence replicates across the microscopic strands of DNA. The human genome contains over 3 billion DNA base pairs enclosed within 23 pairs of chromosomes. On the long arm of the 15th chromosome, a single nucleotide polymorphism occurs at a specific genetic locus named RS129138832. At this exact physical location, on the DNA strand, a thymine molecule physically replaces a cytosine molecule. This microscopic structural change happens inside the specific sequence of the HERC2 gene. The standard sequence of the HERC2 gene functions as a chemical promoter. The genetic sequence binds physically with transcription proteins to activate the neighboring OCA2 gene. The OCA2 gene contains the exact biological instructions for manufacturing the P protein. The P protein builds and transports the melanosomes responsible for depositing brown eumelanin inside the front layers of the human eye. The substitution of the single thymine molecule alters the physical three-dimensional shape of the genetic binding site. The transcription proteins slide past the altered DNA sequence, leaving the neighboring OCA2 gene in a permanently dormant state. The melanocytes located in the anterior border layer and the stroma of the child's eyes halt all production of the P protein. The melanocyte cells completely cease depositing brown eumelanin into the front layers of the eye. The posterior pigment epithelium, the cellular layer at the very back of the iris, continues producing dense black-brown melanin according to an entirely separate set of genetic instructions. The stroma, situated directly in front of this dark layer, develops as a structurally clear tissue. The stroma consists of a fluid matrix containing suspended collagen proteins, fibroblasts, and branching blood vessels. When visible sunlight hits the child's face, the electromagnetic waves pass straight through the transparent cornea and enter the clear stroma. The long wavelengths of light, measuring between 600 and 700 nanometers, travel directly through the front layer of tissue. The solid layer of dark melanin at the back of the iris absorbs these long red and yellow wavelengths completely upon contact. The short wavelengths of light, measuring between 400 and 500 nanometers, strike the microscopic collagen fibers suspended in the stroma fluid. These collagen fibers measure between 10 and 30 nanometers in diameter. The physical collision causes the short blue light waves to scatter outward in multiple directions. Optical physicists classify this specific phenomenon as Tyndall scattering. The scattered blue wavelengths exit the stroma and travel back through the transparent cornea into the surrounding air. The physical structure of the eye generates a vivid blue color entirely through the mechanical scattering of incoming light. The child reaches sexual maturity and reproduces with other members of the agricultural settlement. The individual passes the mutated chromosome 15 down to the next generation of children. The specific genetic sequence for blue eyes spreads through the local population along the Black Sea coast over the next several centuries. The humans living in these coastal settlements migrate inland along the river valleys of Eastern Europe. They carry the mutated HERC2 gene into the surrounding hunter-gatherer populations through interbreeding. The local gene pool expands to include solid brown eyes and pure blue eyes. The DNA replicates strictly across the generations, maintaining the specific cytosine or thymine molecules at the exact RS12913-832 location. Around 5,300 years ago, a new human culture named the Yamnaya emerges on the Pontic-Caspian steppe, a vast grassland stretching from the northern shores of the Black Sea to the Caspian Sea. The Yamnaya pastoralists domesticate the wild horse, Equus caballus, for riding and physical transport. They construct heavy wooden wagons fitted with solid circular wheels carved directly from thick oak tree trunks. They hitch domesticated cattle to these wooden wagons to transport their woolen tents and food supplies across the open plains. The Yamnaya populations carry the specific genetic sequences for lighter skin and light eyes. The mobile herders begin a massive physical expansion westward into the established agricultural zones of Europe, bringing their distinct genetic traits directly into contact with heavily pigmented, dark-eyed populations. The Yamnaya pastoralists drive their wooden wagons westward across the Carpathian Mountains starting in 2900 BCE. They bring herds of domesticated Bos Taurus cattle into the river valleys of Central Europe. The wagons feature solid disc wheels measuring 60 centimeters in diameter, secured to rotating wooden axles with animal hide lashings. The cattle consume the tall fescue grasses growing in the wet alluvial soil. The herders wear thick woolen tunics dyed with red ochre, a natural clay earth pigment containing heavy concentrations of iron oxide. The pastoralists encounter established agricultural communities living in timber longhouses measuring up to 50 meters in length. These settled farmers cultivate emmer wheat and domesticate pigs in the surrounding oak forests. The agrarian communities manufacture ceramic pottery by pressing twisted cords of bast fiber into wet clay before firing it in earthen kilns. The two distinct populations share these agricultural resources and interbreed. The cellular reproduction between these two groups physically combines isolated genetic lineages. During human fertilization, 23 chromosomes from the male sperm fuse with 23 chromosomes from the female egg inside the fallopian tube. The resulting single-cell zygote contains a complete diploid genome of 46 chromosomes. The Yamnaya parents contribute the mutated HERC2 allele, which halts melanin production in the eye. The agrarian parents contribute the standard, functional HERC2 allele, which signals the neighboring OCA2 gene to manufacture large volumes of the P protein. The newly formed genetic sequence places these conflicting biological instructions onto the same homologous chromosome pair at locus 15 CROSQ13. The Bell Beaker culture expands westward across the continent by 2800 BCE, carrying these newly mixed genetic markers into modern-day Spain and Britain. They mine copper ore from surface veins, smelt the metal in clay crucibles heated by charcoal fires, and cast broad-bladed daggers. The human genome utilizes 61 distinct genes to regulate the exact cellular distribution of melanin. When the offspring inherit this specific mixture of active and inactive regulatory alleles, the cellular transcription proteins read a combined heterozygous sequence. The genetic promoters operate at a reduced functional capacity. The OCA2 gene transcribes a strictly limited volume of messenger RNA. The cellular ribosomes translate this messenger RNA into a restricted supply of the P protein. The P protein facilitates the transport of tyrosine molecules inside the melanocyte cells. The synthesis of eumelanin requires a specific chemical environment inside the melanosome. The P protein regulates the potential hydrogen or pH level of the cellular organelle. The restricted volume of P protein maintains a neutral pH balance, which slows the chemical processing speed of the tyrosinase enzymes. The melanosomes mature at a reduced rate and store a lower total volume of the dark pigment before traveling outward along the cellular microtubules. The restricted supply of P protein limits the conversion of tyrosine into brown eumelanin. The melanocytes, located in the anterior border layer of the human iris, receive this limited supply of eumelanin. The cells synthesize a moderate volume of the brown pigment. The melanocytes distribute this pigment asymmetrically across the 12 mm diameter of the iris stroma. They deposit a dense ring of eumelanin directly around the pupillary margin, the inner edge of the iris immediately surrounding the central pupil. The melanocytes leave the outer peripheral zone of the stroma sparsely pigmented. The structural tissue of the stroma consists of suspended collagen proteins and a network of branching capillaries pumping oxygenated blood. Ambient sunlight enters the anterior chamber of the eye. The dense pigment ring near the pupil absorbs the light rays, creating a visible ring of brown around the center of the eye. In the sparsely pigmented outer edges, the incoming light hits the suspended microscopic collagen fibers. The physical collision scatters the short blue wavelengths outward through Tyndall scattering. The scattered blue light intersects with the sparse clusters of brown eumelanin suspended in the outer stroma. The physical overlapping of the scattered blue wavelengths and the brown pigment produces the visual wavelength of green, measuring between 500 and 550 nanometers. The biological combination of the brown central ring and the scattered green periphery establishes the physical structure of a hazel eye. These intermediate genetic traits spread through the European continent over the next millennium. The populations expand south along the Balkan Peninsula. They navigate the Aegean Sea using wooden rowboats constructed from cut pine timber sealed with heated tree resin. They settle in the coastal valleys of the Peloponnese region. By 1600 BCE, these populations establish the Mycenaean civilization, building fortified palaces out of heavy limestone blocks on the high rocky hills of southern Greece. The citizens of these settlements cultivate olive trees in the dry, rocky soil and press the fruit to extract oil. They transport this olive oil in terracotta amphorae across the Mediterranean Sea, trading with the Minoan civilization on the island of Crete and the established dynasties of the Egyptian Old Kingdom. The maritime trade routes facilitate constant genetic exchange between the diverse coastal populations. Merchant sailors carrying the genetic markers for light and intermediate eye colors intermarry with populations carrying the ancient sequences for heavy melanin production. The regional gene pool absorbs the 61 separate regulatory genes into a shared biological network. The genetic sequences for moderate melanin production multiply across the Mediterranean basin through continuous population growth. The human gene pool in the region stabilizes into a highly diverse mixture of phenotypes. Individuals with dark brown eyes, pure blue eyes, and hazel eyes inhabit the exact same geographic settlements. By 350 BCE, the descendants of these populations build densely populated urban city-states across the Greek peninsula. The citizens display this wide spectrum of eye colors, prompting local scholars to actively record the physical traits of the families living in the surrounding architecture. The year is 350 BCE. The location is the city of Athens, situated on the Attica Peninsula near the Aegean Sea. The local temperature rests at 24 degrees Celsius. The sun illuminates the dirt pathways of the Lyceum, a public gymnasium and meeting space located just outside the eastern limestone walls of the city. The architectural space contains rows of fluted marble columns measuring 10 meters in height. A warm wind blows dry red dust across the stone paving blocks. The philosopher Aristotle walks along these open pathways. He wears a heavy himation draped over his left shoulder. The natural wool smells of lanolin and wood smoke. Aristotle carries a sheet of dried papyrus, imported from the Egyptian city of Alexandria. The papyrus consists of plant stalks, sliced into thin strips, soaked in water, and pressed together under heavy stones to form a flat, biological writing surface. He holds a calamus, a writing pen carved from a dried marsh reed. He dips the split tip of the reed into a terracotta inkwell. The inkwell contains a black liquid manufactured by mixing carbon soot, water, and sticky Arabic gum harvested from acacia trees. Aristotle sits on a curved marble bench. The marble physically absorbs the ambient heat of the midday sun. He places the papyrus on his lap and applies the carbon ink to the plant fibers. He writes the Greek text, Generation of Animals. He records his direct physical observations of the human families living in the surrounding stone houses of Athens. He documents the exact physical inheritance of anatomical traits across multiple generations of citizens. He interviews local men and women, directly recording the physical eye colors of the biological parents and the resulting eye colors of their offspring. He catalogues the human eyes into distinct categories based on their visible color under natural sunlight. He writes the Greek word melas to describe dark brown eyes. These dark eyes contain dense concentrations of brown pigment. He writes the word glaukos to describe blue or gray eyes. These lighter eyes scatter the short blue wavelengths of incoming sunlight. He writes the word karapos to describe an intermediate eye color. The Greek term karapos directly translates to a yellowish-brown or mixed biological trait. He uses this specific word to categorize irises displaying a physical blend of dark brown pigment and scattered blue light. Optometrists today classify this exact intermediate biological phenotype as Hazel. Aristotle observes and records the physical presence of this specific, mixed trait within the Athenian population. Aristotle records multiple instances where human children possess eye colors completely distinct from their immediate biological parents. He documents a family where a male with melas, dark brown eyes, reproduces with a female possessing glaukos, blue eyes. He notes the resulting offspring develops karopos, hazel eyes. He writes these exact observations into the 5th book of his manuscript. He records that biological material from both parents physically mixes inside the human body during sexual reproduction. He continues his observations, noting that physical traits routinely skip entire generations. He records a specific lineage featuring a grandparent with hazel eyes, passing the trait directly to a grandchild, while the intermediate parent displays strictly dark brown eyes. He documents the visible results of epistasis, the biological process where multiple genes interfere with and modify each other's expressions. He records the physical mechanics of this heredity 22 centuries before the scientific discovery of genetic sequences and microscopic chromosomes. Aristotle expands his manuscript by documenting the physical structure of the animal and human eye. He dissects the eyes of sheep and goats using a sharp bronze blade. He notes the clear fluid inside the anterior chamber of the eye. He observes the iris tissue sitting directly behind the transparent cornea. He records the physical contraction of the central pupil when exposed to direct sunlight, noting the mechanical movement of the iris tissue. He documents the visible structural color of human infants living in the city. He records that the majority of human infants born in Athens enter the world with glaukos, blue eyes. He observes these infants over a period of 36 months. He writes that the infant eye color physically darkens during the first several years of life. He notes the color transitions from blue to charopos or melas as the child ages. He documents the exact biological timeline of ocular melanocytes slowly accumulating eumelanin in the stroma, a process requiring direct light exposure to activate the cellular machinery. Aristotle attempts to explain the physical cause of these shifting colors using the available scientific theories of his era. He hypothesizes that the physical volume of fluid inside the eyeball dictates the final visible color. He writes that a deep volume of fluid creates dark colors, and a shallow volume of fluid creates light colors. He applies the carbon ink to the papyrus, writing in continuous Greek uncial script and leaving 0 spaces between the individual words. He finishes the chapter rolls the papyrus sheet into a tight cylinder, and stores it inside a circular leather capsule. Scribes physically copy Aristotle's manuscript onto fresh animal parchment pages over the next 2 millennia. They scrape sheepskins with iron pumice stones to create flat writing surfaces. They use sharpened bird feathers to transcribe the Greek observations. The physical text travels along maritime trade routes through the Byzantine Empire and the Islamic Golden Age. Scholars sitting in the libraries of Baghdad and Toledo translate the original Greek words into Arabic and Latin manuscripts. They bind the parchment pages between wooden boards covered in tooled leather. The observational data regarding intermediate eye colors and genetic inheritance remains the scientific standard in European universities for 2,000 years. Medical students in Padua and Paris memorize Aristotle's categories of melas, glaucos, and charopos. The human eye lacks the optical magnification required to observe the exact biological substance responsible for these shifting colors. The physical search for this microscopic pigment requires advanced optical glass lenses. This search shifts to the anatomical laboratories of Western Europe in the late 19th century. The year is 1873. The location is the Faculty of Medicine in Paris, France. The laboratory sits inside a stone building on the Rue de l'École de Médecine. The ambient temperature inside the room is 18 degrees Celsius. French anatomist Charles-Philippe Robin stands beside a heavy oak table. He wears a starched cotton laboratory coat, smelling of carbolic acid and ethyl alcohol. He places a brass compound microscope on the wooden surface. The microscope uses 2 precisely ground glass lenses to magnify microscopic biological structures. The lower objective lens gathers light, passing directly through a cellular sample. The upper eyepiece lens magnifies the resulting image up to 400 times its original size. A flat, silver mirror sits on a rotating metal hinge at the base of the instrument. Robin adjusts the physical angle of the mirror to catch the natural sunlight entering through a tall glass window. The mirror reflects the sunlight upward through a circular aperture in the stage and directly into a glass slide. Robin prepares this slide using human skin tissue extracted from a cadaver. He uses a steel microtome blade to slice a cross-section of the epidermis, measuring exactly 5 micrometers in thickness. He uses metal forceps to lay the tissue flat against the rectangular glass base. He adds a single drop of distilled water over the tissue sample. He places a thin, square glass cover slip over the water, pressing the liquid flat to eliminate trapped air bubbles. Robin positions his eye over the brass eyepiece and turns a metal dial on the side of the microscope barrel. The dial physically lowers the lens closer to the glass slide until the cellular structures come into sharp focus. He observes the individual keratinocyte cells forming the thickest layers of the epidermis. Inside the basal layer of the skin, he observes distinct, dark brown granular particles accumulating densely around the cellular nuclei. He writes his direct observations onto a sheet of paper using a steel-nibbed fountain pen filled with iron gall ink. He uses the term melanin to classify this specific dark pigment in his notes. He derives the word directly from the Greek word melas, meaning black. The exact term Aristotle used 22 centuries earlier. Robin publishes these structural findings in his medical text Anatomie et physiologie cellulaire. The publication formally establishes the biological classification of this pigment in European medical literature. Later analytical chemists isolate the specific chemical compounds making up this granular pigment. They divide human melanin into 2 distinct molecular structures based on their chemical composition. They name the first structure eumelanin. Eumelanin molecules consist of tightly cross-linked polymers containing carbon, hydrogen, nitrogen, and oxygen atoms. The dense physical structure of eumelanin absorbs large amounts of ultraviolet radiation. The chemical compound appears deep brown or black under visible light. The chemists identify a second molecular variation and name it pheomelanin. The melanocytes synthesize pheomelanin by incorporating the amino acid cysteine into the polymer chain during the initial stages of cellular oxidation. The cysteine adds heavy sulfur atoms to the basic chemical structure. The physical presence of sulfur alters the absorption of incoming electromagnetic light, causing pheomelanin to reflect long red and yellow visible wavelengths. The human body deposits pheomelanin in high concentrations in red hair follicles, the cellular tissue of the lips, and the specialized skin of the areola. The chemical stability of pheomelanin degrades significantly faster than eumelanin when exposed directly to ultraviolet radiation, releasing free radicals into the surrounding skin tissue. Cellular biologists extract tissue samples from the human eye to determine the exact distribution of these two specific pigments inside the iris. They place cross-sections of human iris tissue under high-powered electron microscopes. They apply chemical stains to isolate the specific melanin variants within the cellular matrix. The chemical analysis reveals that human eye color relies exclusively on the concentration of brown eumelanin. The melanocytes operating inside the stroma of a hazel eye produce strictly eumelanin. The final visible hue of the hazel iris depends entirely on the precise physical volume of the brown eumelanin molecules suspended in the anterior tissue. The biological process of building this pigment occurs inside the melanocyte cell. The cell imports the amino acid tyrosine directly from the human bloodstream through the cellular membrane. Inside the membrane-bound melanosome organelle, an enzyme called tyrosinase catalyzes a chemical reaction, physically stripping electrons from the tyrosine molecules. The chemical oxidation converts the clear tyrosine into a compound called dopaquinone. The dopaquinone molecules spontaneously bond to each other, building the large, light-absorbing polymer chains of eumelanin. The melanocyte cell utilizes motor proteins to transport the mature melanosomes along a network of microscopic actin filaments. These motor proteins move the pigment outward into the long, branching dendrites of the cell. In a hazel eye, the tyrosinase enzyme processes a strictly limited volume of tyrosine due to the specific genetic instructions from the mutated chromosome 15. The melanosomes mature with a low density of eumelanin polymer chains. The melanocytes push these partially filled melanosomes out into the fluid matrix of the stroma. The melanin sits in sparse clusters among the collagen fibers. The pigment clusters measure between 300 and 500 nanometers in diameter. The structural tissue of the stroma contains high concentrations of water and hyaluronic acid, creating a thick, transparent fluid suspension for the pigment molecules, the structural proteins, and the branching blood vessels. The physical distance between these sparse pigment clusters leaves wide sections of the collagen fibers completely exposed to incoming ambient light. Visible sunlight enters the eye, traveling at a speed of 299,792 kilometers per second. The electromagnetic light waves pass around the sparse brown pigment clusters and strike the bare collagen proteins directly. The specific physical interaction between the incoming electromagnetic radiation and these microscopic protein fibers dictates the resulting visual color of the eye. The physical collision between the white sunlight and the suspended particles forces specific, high-frequency wavelengths to alter their trajectory and scatter outward into the surrounding space. The year is 1869. The location is the Royal Institution of Great Britain on Albemarle Street in London, England. The laboratory room sits at a temperature of 15 degrees Celsius and smells of burning coal and ozone. Irish physicist John Tyndall stands beside a heavy mahogany optical workbench measuring 2 meters in length. He wears a dark wool suit jacket and manipulates a series of cylindrical glass tubes. Tyndall generates a bright light using a carbon arc lamp. The lamp consists of 2 pointed rods of solid carbon connected to a copper wire dynamo. He brings the tips of the carbon rods within 2 millimeters of each other. The electrical current leaps across the physical gap, ionizing the air molecules and creating a glowing plasma arc operating at 3,600 degrees Celsius. The carbon arc lamp emits a continuous, high-intensity beam of broad-spectrum white electromagnetic radiation. Tyndall mounts a brass collimator lens directly in front of the plasma arc. The convex glass lens focuses the scattered light rays into a single, parallel beam traveling in a straight horizontal line. He places the glass tubes onto brass stands directly in the path of this concentrated light beam. He fills the first cylindrical glass tube with distilled water. The white light enters the clear water, passes completely through the liquid medium, and strikes the flat plaster wall behind the optical bench. The light leaves a solid white circular illumination on the painted surface. Tyndall prepares a second fluid medium. He grinds a piece of solid mastic resin into a fine powder, using a marble mortar and pestle. Mastic resin consists of organic polymers harvested from the bark of the Pistachia lentiscus tree native to the Mediterranean region. He drops microscopic particles of this yellow resin into the pure water. The resin particles remain suspended in the fluid, creating a turbid, cloudy mixture. Tyndall directs the focused white light beam into this cloudy liquid. The visible spectrum of white light contains distinct electromagnetic wavelengths ranging from 400 nanometers to 700 nanometers. The longer wavelengths, measuring between 600 and 700 nanometers, correspond to the visible color red. These long red waves pass smoothly around the microscopic resin particles and exit through the back of the glass tube to hit the plaster wall. The shorter wavelengths, measuring between 400 and 500 nanometers, correspond to the visible color blue. These short blue electromagnetic waves collide physically with the solid resin particles suspended in the water. The mechanical collision deflects the short blue waves off their linear path. The photons scatter laterally across the darkened laboratory, illuminating the sides of the cylindrical glass tube with a bright blue hue. Tyndall measures the angle and intensity of the scattered light using a specialized optical instrument called a goniometer. He records these specific optical measurements in his laboratory notebook using a steel-nibbed dip pen. He publishes his findings in the Philosophical magazine, establishing the formal scientific principles of the Tyndall effect. Medical researchers apply the physical principles of the Tyndall effect directly to the microscopic structures of the human eye. The human iris relies entirely on this specific form of structural scattering to generate cool-toned visual frequencies. The human body constructs the stroma layer of the iris using a complex extracellular matrix. Fibroblast cells excrete chains of amino acids, weaving together dense fibers of type 1 and type 6 collagen. These microscopic collagen strands measure between 10 and 30 nanometers in physical diameter. The fibroblasts suspend these protein strands inside a thick fluid composed of water dissolved sodium salts, and hyaluronic acid molecules. Ambient daylight, traveling at 299,792 kilometers per second, enters the front of the eye. The broad-spectrum white light passes through the transparent cornea and enters the fluid matrix of the stroma. The long red and yellow wavelengths push through the fluid, passing around the thin collagen fibers. These long waves reach the posterior pigment epithelium, located at the very back of the iris structure. The solid, two-cell-thick layer of dense black-brown eumelanin absorbs the long red and yellow wavelengths entirely, converting the electromagnetic energy into microscopic fractional amounts of heat. The short blue wavelengths within the daylight spectrum hit the suspended collagen strands in the anterior stroma. The physical dimension of the collagen fibers matches the exact necessary parameters to trigger Tyndall scattering. The blue photons ricochet off the protein strands, scattering forward and outward through the cornea. This mechanical scattering generates the physical appearance of blue color, utilizing strictly transparent biological tissues and clear extracellular fluid. The specialized melanocytes in a hazel eye introduce one modifying variable into this optical system. The cells deposit a moderate volume of brown eumelanin clusters directly among the scattering collagen fibers. The scattered blue photons bounce off the collagen and immediately intersect with the surrounding brown pigment granules. The visual spectrum of the scattered blue light physically mixes with the yellow and brown light waves reflected by the sparse eumelanin. This optical combination produces hybrid wavelengths measuring specifically between 500 and 550 nanometers. The human retina captures these specific wavelengths using photoreceptor cone cells located at the back of the eyeball. The optic nerve transmits the electrical signals to the occipital lobe at the rear of the human brain. The visual cortex processes this specific frequency combination and registers the resulting color as green. The cellular distribution of the brown pigment varies randomly across the total 12 mm diameter of the iris. Areas containing denser clusters of eumelanin absorb more light, creating visible patches of amber and gold. Areas containing widely spaced melanin granules allow the Tyndall scattering to dominate, producing visible patches of structural green. The exact spatial arrangement of the melanin granules determines the final visible color and texture of the hazel eye. The ratio of scattered blue light to absorbed red light shifts dynamically based on the microscopic placement of every individual pigment cluster. To map this complex physical geometry, ocular researchers extract whole cross-sections of the human iris tissue for specialized microscopic examination. Histologists operating inside modern clinical laboratories utilize heavy metal chemical stains and high-voltage electron microscopes to chart the exact cellular architecture holding these suspended pigment particles. They slice the biological tissue into layers measuring only 1 micrometer in thickness to measure the precise anatomical distance between the front border layer and the dark posterior epithelium. The year is 1905. The location is the Institute of Anatomy at the University of Leipzig in the German Empire. The laboratory room maintains a steady air temperature of 20 degrees Celsius. The atmosphere inside the room smells of formaldehyde, ethanol, and cedarwood oil. German histologists extract a complete human eye from a biological specimen. They place the spherical eyeball, measuring exactly 24 millimeters in diameter, into a glass beaker. The beaker contains a 10% formalin solution. The formaldehyde molecules cross-link the cellular proteins, physically hardening the biological tissue. The histologists use a steel scalpel to sever the clear cornea from the front of the eye. They extract the intact iris. The human iris forms a flat, circular ring measuring exactly 12 millimeters in outer diameter and 0.5 millimeters in thickness. They wash the tissue in increasing concentrations of ethanol liquid, starting at a 50% mixture and ending at 100% pure alcohol. The alcohol physically extracts the water molecules from the cellular walls. They submerge the dehydrated tissue in liquid xylene, a clear hydrocarbon solvent, before embedding the circular tissue into a block of liquid paraffin wax, heated to 60 degrees Celsius. The wax cools and solidifies around the biological sample over a period of 2 hours. A laboratory technician mounts the solid wax block onto a heavy iron rotary microtome. The technician rotates a steel handwheel, dragging a sharp metal blade across the wax block. The blade shears off continuous biological tissue sections, measuring exactly 5 micrometers in thickness. The technician places these microscopic slices onto rectangular glass slides, measuring 25 by 75 millimeters. They apply a chemical stain called hematoxylin directly to the tissue. The hematoxylin molecules bind to the acidic DNA inside the cellular nuclei, dyeing the biological structures a dark blue color. They apply a second liquid stain called eosin, which dyes the structural proteins and smooth muscle fibers a bright pink hue. The histologists place the stained slides onto the mechanical stage of a Carl Zeiss binocular compound microscope. The brass instrument features apochromatic objective lenses. The specialized glass lenses physically correct chromatic aberration, aligning the red, green, and blue light waves onto a single focal plane. The ocular lenses magnify the tissue section 400 times. The histologists observe the physical cross-section of the iris tissue. They map the exact anatomical structure from the back of the iris to the front. At the absolute rear of the iris, they identify the posterior pigment epithelium. This specific anatomical layer measures exactly 2 cells in total thickness. The cells in this layer form a tight, continuous physical barrier across the entire back surface of the iris. Inside these epithelial cells, the human body packs absolute maximum concentrations of black-brown eumelanin. The melanin granules fill the cellular cytoplasm entirely, forcing the blue and pink chemical stains to remain on the exterior of the cells. This opaque, black-brown layer exists at the back of every human iris. The dark pigment absorbs incoming light that penetrates deep into the eye. The melanin converts the photon energy into microscopic heat, shielding the internal ocular structures from severe thermal damage. Directly in front of this posterior layer, the histologists document a second cellular barrier named the anterior pigment myoepithelium. The basal sections of these specialized cells extend smooth muscle fibers radially toward the outer edge of the iris. These extended muscle fibers form the dilator pupillae muscle. The sympathetic nervous system sends electrical signals to these fibers, causing them to physically contract and pull the central pupil open. Moving forward from the muscular layers, the histologists examine the thickest section of the iris, named the stroma. The stroma consists of a loose matrix of connective tissue. Fibroblast cells construct a physical scaffold using type 1 and type 6 collagen proteins. Microscopic capillaries branch through this collagen network, pumping oxygenated blood to the surrounding cells. Near the central pupillary margin, smooth muscle fibers form a dense, circular ring called the sphincter pupillae. The parasympathetic nervous system triggers this circular muscle to constrict, physically reducing the diameter of the pupil. At the very front of the stroma, directly facing the clear cornea, the tissue condenses into the anterior border layer. This thin layer contains a dense network of specialized melanocyte cells. The histologists document oval-shaped pits penetrating the front surface of this layer. These physical indentations, called Fuchs crypts, measure 1/10 of a millimeter in width. The open crypts allow the aqueous humor fluid to flow directly from the anterior chamber into the deep stroma tissue as the iris muscles expand and contract. In a hazel eye, the melanocytes residing in the anterior border layer manufacture a moderate volume of brown eumelanin. The cells distribute this pigment asymmetrically across the upper sections of the stroma. The melanin clusters sit suspended in the extracellular fluid, separated by wide gaps of clear collagen fibers. The individual melanin granules measure exactly 0.8 micrometers in diameter. When a living human opens their eyelid, broad-spectrum sunlight hits the clear cornea. The electromagnetic radiation travels through the liquid aqueous humor and strikes the front surface of the anterior border layer. The photons pass directly between the scattered clusters of brown eumelanin and hit the bare collagen fibers inside the stroma. The physical collision with the collagen strands scatters the short blue wavelengths outward. The long red and yellow wavelengths continue straight through the stroma fluid. The two-cell-thick posterior pigment epithelium at the back of the iris absorbs these long wavelengths completely. The scattered blue light waves, measuring exactly 450 nanometers, travel back out through the front of the eye. Before exiting the stroma, these blue photons physically intersect with the localized clusters of brown eumelanin. The brown pigment reflects a distinct percentage of yellow light waves, measuring 570 nanometers, outward into space. The blue wavelengths and the yellow wavelengths cross physical paths in the space directly in front of the human eye. The biological blending of these distinct electromagnetic frequencies produces the visual hue of a hazel iris. The total visible color of the eye relies entirely on the macroscopic distribution of this pigment across the tissue. The melanocytes deposit heavy concentrations of pigment in specific geographic zones of the iris, depositing only clear fluid and bare collagen fibers in the remaining zones. This asymmetrical physical variation in melanin density creates distinct geographic shapes visible to the naked human eye. Optometrists map these specific pigment distributions to categorize the macroscopic geometric structures of human vision. The year is 1955. The location is a clinical examination room inside the School of Optometry at the University of California, Berkeley. The room temperature sits at 21 degrees Celsius. The air smells of isopropyl alcohol used to sterilize clinical equipment and warm metal emanating from electrical light fixtures. An optometrist sits on a specialized stool equipped with hydraulic height adjustment. The patient sits on the opposite side of a motorized instrument table. The optometrist presses a circular metal button, activating an electric hydraulic pump that raises the heavy wooden table to exactly chest height. The optometrist utilizes a slit-lamp biomicroscope to examine the macroscopic physical geometry of the patient's eye. The slit lamp consists of a heavy cast-iron base supporting a complex optical viewing system and a mechanical illumination arm. The patient places their mandible into a molded plastic chin rest and presses their frontal bone against a curved rubber forehead strap. This physical restraint completely stabilizes the human head. The optometrist activates the illumination system. A flow of electrical current travels through copper wires to a tungsten halogen incandescent bulb. The electrical resistance heats the tungsten filament to 2,800 degrees Celsius. generating a continuous beam of broad-spectrum white electromagnetic radiation. The bulb housing features an interior coating of black enamel paint to absorb scattered photons. The white light passes through a series of internal glass condenser lenses and hits a mechanical aperture. The optometrist turns a knurled brass dial, sliding two metal plates together to form a rectangular opening measuring exactly 0.2 millimeters in width and 8 millimeters in height. This mechanical restriction shapes the light into a focused, vertical rectangular beam. The beam exits the housing and strikes the transparent cornea of the patient. The optometrist leans forward and looks through two Galilean binocular eyepieces. The internal apochromatic glass lenses magnify the illuminated biological tissue 40 times its natural size. The optical magnification reveals the exact topographical surface of the human iris. The optometrist documents the strict geographic distribution of the brown eumelanin pigment across the 12 mm diameter of the hazel eye. The human iris divides physically into two distinct concentric anatomical zones. The inner region, located closest to the central opening of the pupil, is the pupillary zone. The outer region, extending to the white sclera of the eyeball, is the ciliary zone. A jagged, circular ridge called the collarette physically separates these two regions. The collarette sits exactly 1.5 millimeters outward from the inner pupillary margin. The collarette marks the thickest physical section of the iris tissue. This raised ridge consists of overlapping bands of type 1 collagen fibers and connective extracellular matrix. The ridge marks the exact anatomical location where an embryonic vascular network existed during the fetal development of the human body. In a hazel eye, the melanocyte cells deposit the absolute highest concentration of brown eumelanin pigment within the pupillary zone and directly along the physical ridge of the collarette. The dense microscopic clusters of brown pigment pack tightly together in this inner geographic circle. When the rectangular beam of white light hits this central zone, the high volume of eumelanin absorbs the incoming electromagnetic wavelengths, measuring between 400 and 700 nanometers. The biological absorption converts the light energy into microscopic heat. The physical tissue reflects a distinct, solid brown color directly surrounding the central pupil. The melanocytes deposit significantly lower volumes of eumelanin pigment in the outer ciliary zone. The tissue extending from the collarette outward to the edge of the iris consists largely of bare collagen fibers and clear stromal fluid. The collarette itself features an irregular, zigzag physical shape extending outward into this clearer tissue. Microscopic streaks of brown pigment travel outward from the central ridge, following the structural alignment of the underlying collagen cords. These physical extensions of brown pigment create a geometric radiating pattern extending outward from the center of the eye. Clinical observers document this specific physical arrangement as a sunburst or starburst anatomical pattern. The low density of brown melanin in the outer ciliary zone exposes the suspended collagen fibers to the incoming white light. The physical collision between the light and the collagen triggers Tyndall scattering. The protein strands scatter the short blue wavelengths, measuring exactly 450 nanometers outward in random directions. The scattered blue light waves intersect with the extremely sparse brown pigment clusters deposited at the outer edge of the sunburst pattern. The physical overlapping of the scattered blue photons and the yellow wavelengths reflected by the sparse eumelanin produces a structural green hue in the ciliary zone. The complete macroscopic geometry of the hazel eye consists of the central brown ring contained within the collarette, the radiating brown extensions forming the sunburst, and the structural green of the outer ciliary periphery. The optometrist documents deep radial furrows cutting through the anterior border layer of the ciliary zone. These furrows consist of physical creases in the cellular tissue, forming concentric arcs near the outer edge of the iris. The parasympathetic nervous system sends an electrical impulse to the sphincter pupillae muscle located inside the stroma. The circular muscle physically contracts, pulling the pupillary zone inward and reducing the diameter of the central pupil. This mechanical contraction stretches the iris tissue across the anterior chamber. The physical stretching pulls the radial furrows flat and alters the exact physical distance between the microscopic brown eumelanin clusters in the sunburst pattern. The geometric expansion changes the surface area of the exposed collagen fibers. The shifting tissue physically alters the ratio of scattered blue light to absorbed red light, shifting the exact visible color of the hazel eye under the slit lamp beam. The optometrist records these specific dynamic structural changes in a paper medical file using a graphite pencil. During the exact same decade, in the 1950s, the American educational system standardizes the biology curriculum across thousands of public high schools. Publishing companies in New York and Chicago operate massive offset lithography printing presses. Giant cylindrical steel rollers apply carbon-based black ink to continuous rolls of white wood pulp paper. The publishers mass-produce millions of identical biology textbooks for the national student population. The educational curriculum mandates the instruction of human genetic inheritance. The textbooks print structured grid diagrams utilizing the 19th century agricultural research of an Augustinian friar named Gregor Mendel. The year is 1958. The date is September 15th. The location is a biology classroom inside Oak Park and River Forest High School in the state of Illinois. The room temperature measures exactly 22 degrees Celsius. The air smells of paste wax applied to the linoleum floor and dry calcium carbonate dust floating in the ventilation currents. The classroom contains 30 wooden desks constructed from varnished oak timber. The desks feature cast iron legs bolted directly into the floor joists. A public school teacher stands in front of a rectangular slab of dark slate rock mounted to the front wall. The teacher holds a cylindrical piece of white chalk measuring exactly 10 centimeters in length. The chalk consists of compressed calcium carbonate, a mineral harvested from sedimentary limestone deposits. The teacher presses the white mineral against the dark slate, applying physical friction. The friction shears off microscopic particles of the chalk, leaving a continuous white line across the stone surface. The teacher draws a square frame containing one vertical line and one horizontal line intersecting at the exact center. This physical diagram creates a grid with 4 equal quadrants. Biologists classify this specific mathematical tool as a Punnett square. British geneticist Reginald Punnett developed this visual matrix in 1905 to calculate the mathematical probability of inherited physical traits. The students sit at the wooden desks. They read from a standardized textbook titled Modern Biology, originally authored by Truman J. Moon and published by Henry Holt and Company. The publishing plant prints the text on offset lithography presses, pressing carbon-based black ink into the porous fibers of white wood pulp paper. The textbook devotes an entire chapter to the agricultural experiments conducted by Gregor Mendel in 1865. Mendel operated an experimental greenhouse in the garden of the St. Thomas Abbey in the city of Brno. Between 1856 and 1863, he cultivated and tested exactly 28,000 plants of the species Pisum sativum, the common garden pea. He manually transferred pollen between the reproductive organs of the plants using a small camel hair paintbrush. He tracked 7 distinct physical traits across multiple generations, including seed shape, pod color, and stem length. Mendel observed that certain physical characteristics completely masked the visual appearance of other characteristics. He labeled the visible traits as dominant and the hidden traits as recessive. The biology curriculum in 1958 applies this exact 19th century agricultural model directly to human eye color. The teacher writes a capital letter B in the top margin of the slate board to represent the genetic instruction for brown eumelanin pigment. The teacher writes a lowercase letter b to represent the genetic instruction for a lack of pigment resulting in blue eyes. The educational framework instructs the students that human eye color functions exclusively as a monogenic trait. This biological classification dictates that a single specific gene controls the entire physical outcome of the iris color. The teacher fills the 4 quadrants of the chalk grid. The mathematical model demonstrates the reproductive crossing of 2 heterozygous parents. Each parent carries 1 capital B allele and 1 lowercase b allele. The teacher calculates the probabilities by combining the letters in the 4 boxes. The completed grid displays 1 double capital B, 2 mixed capital and lowercase boxes, and 1 double lowercase b. The teacher instructs the class that the capital letter always masks the lowercase letter. The final mathematical calculation yields a 75% probability of producing a child with solid brown eyes and a 25% probability of producing a child with solid blue eyes. The students copy the grid onto ruled notebook paper using graphite pencils. The printed textbook and the slate diagram limit human eye color to strict binary categories. The educational materials exclusively feature solid brown and solid blue eyes. The complex geometric structure of a hazel eye, featuring moderate melanin concentrations and scattered blue light waves, exists entirely outside the parameters of the monogenic Punnett square. The textbook provides 0 genetic explanation for the radiating sunburst pattern of brown pigment surrounded by green collagen fibers. The American educational system utilizes this simplified dominant recessive model for human eye color inheritance for the next 40 years. Generations of students memorize the binary charts to pass state-mandated, standardized examinations. As clinical record-keeping expands in hospitals during the late 20th century, medical professionals document thousands of physical anomalies that disrupt the single-gene theory. Biological parents possessing pure blue eyes frequently produce children with hazel irises. The strict mathematical rules of the classroom Punnett square classify this specific physical outcome as a biological impossibility. Genetic researchers eventually discard the single-gene agricultural model for human eye color. They adopt advanced molecular biology techniques to map the actual physical structure of human inheritance at the microscopic level. Phlebotomists extract whole blood samples from human patients using hollow stainless steel needles. Laboratory technicians place the blood vials into centrifuge machines. An electric motor spins the vials at 3,000 revolutions per minute, using centrifugal force to separate the heavy red blood cells from the lighter white blood cells. The scientists extract pure DNA molecules from the nuclei of these white blood cells. They utilize specialized restriction enzymes to slice the long DNA polymers into smaller, analyzable fragments. In 1990, an international consortium of scientists initiates the Human Genome Project. The researchers operate automated capillary electrophoresis machines to read the exact chemical sequence of the 3 billion nucleotide base pairs forming the complete human genome. The machines utilize lasers to scan fluorescent dyes attached to the genetic fragments, recording the data directly into digital computer mainframes. On April 14, 2003, the scientists finalize the sequence and isolate the specific genetic control centers dictating melanin production. They locate these primary controls on the microscopic biological structure known as chromosome 15. The date is April 15, 2003. The location is the Wellcome Trust Sanger Institute in Hinxton, England. The data center operates within a secure room featuring a raised floor and active thermal management. The room temperature sits strictly at 18 degrees Celsius. The ventilation system pushes chilled air constantly over 40 steel server racks. The air smells of heated copper wiring and metallic ozone. Researchers sit at workstations, observing liquid crystal display monitors. The illuminated screens display the complete, newly mapped nucleotide sequence of human chromosome 15. The human body constructs chromosome 15 as an acrocentric physical structure. The centromere, the physical constriction point joining two sister chromatids together, sits very close to one end of the biological structure. This asymmetrical placement creates a very short P arm and a long Q arm. Chromosome 15 contains exactly 101,991,189 nucleotide base pairs. This single chromosome represents slightly more than 3% of the total cellular DNA inside a human cell. The physical DNA molecules wrap tightly around octamers of histone proteins, forming dense structural units called nucleosomes. These nucleosomes coil continuously into thick chromatin fibers measuring exactly 30 nanometers in diameter. This precise physical compression allows the entire 3 billion base pairs of the human genome to fit inside a cell nucleus measuring only 6 micrometers in width. To isolate the genetic instructions controlling human eye color, the geneticists pinpoint a specific geographic band located on the long Q arm of chromosome 15. They classify this exact cytogenetic location as 15q13. Laboratory technicians map these locations by applying a chemical mixture called Giemsa stain directly to the chromosome. The stain consists of methylene blue, eosin, and azure B chemicals dissolved in an alcohol solvent. The dye molecules bind strictly to regions of the DNA helix containing high concentrations of adenine and thymine. Adenine and guanine classify as purines, featuring a double-ring chemical structure containing nitrogen and carbon atoms. Cytosine and thymine classify as pyrimidines, featuring a single-ring chemical structure. The Giemsa chemicals bind to the double hydrogen bonds connecting the adenine and thymine molecules. This chemical binding produces visible dark and light horizontal bands across the physical structure of the chromosome under a light microscope. The cytogenetic designation 15q13 corresponds to a specific dark band on the long arm. Within this microscopic geographic band, the DNA sequence contains 2 neighboring genes responsible for the structural coloration of hazel eyes. The scientists identify the first sequence as the OCA2 gene. They identify the adjacent sequence as the HERC2 gene. The HERC2 gene sits directly upstream of the OCA2 gene on the continuous DNA double helix. The DNA strand separates these two specific genes with a physical distance of exactly 2 million nucleotide base pairs. These intervening base pairs constitute an intergenic region containing structural regulatory sequences. The researchers at the Sanger Institute mapped the exact chemical spelling of these genes using automated capillary electrophoresis machines. An electric pump injects a liquid polymer gel into microscopic silica glass tubes. These glass capillaries feature an internal diameter of exactly 50 micrometers. The laboratory technicians load fluorescently tagged DNA fragments from chromosome 15 into the machine. An electrical voltage of 10,000 volts passes through the polymer gel. The strong electrical charge physically pulls the negatively charged DNA fragments through the silica tubes. The smaller DNA fragments move through the thick polymer matrix faster than the larger fragments, organizing the pieces strictly by their physical length. An argon ion laser, positioned at the end of the capillary, shoots a beam of light measuring exactly 488 nanometers in wavelength. The focused green laser beam hits the fluorescent chemical tags attached to the passing DNA molecules. The physical collision of the photons causes the tags to emit specific wavelengths of colored light. A charge-coupled device camera records the specific wavelength of each emitted photon. A computer algorithm translates these light frequencies into a continuous text document consisting of 4 letters, A, C, T, and G. The mapped genetic sequence reveals the precise molecular instructions for human pigmentation. The OCA2 gene contains exactly 344,970 base pairs. The sequence divides physically into 24 distinct coding regions called exons. The neighboring HERC2 gene contains exactly 223,392 base pairs. The sequence divides into 93 separate exons. For a hazel eye to develop in a human infant, the melanocyte cells must access a highly specific combination of these exact base pairs to calibrate a moderate volume of eumelanin. The biological process begins inside the nucleus of the melanocyte cell, located in the stroma of the human iris. An enzyme complex named RNA polymerase II binds physically to the promoter region of the DNA strand at locus 15q13. The enzyme physically severs the hydrogen bonds holding the two strands of the double helix together. The RNA polymerase II moves along the exposed sequence of the OCA2 gene. It matches free-floating ribonucleotide molecules to the exposed DNA bases, bonding cytosine to guanine and uracil to adenine. The chemical bonding constructs a single, continuous strand of messenger RNA. The newly formed messenger RNA strand detaches from the chromosome. The DNA double helix reforms its hydrogen bonds and twists back into its tightly coiled physical shape. The messenger RNA strand travels through the nucleoplasm fluid and exits the nucleus through a cylindrical nuclear pore complex embedded in the nuclear envelope. The messenger RNA enters the cytoplasm of the melanocyte cell. The cytoplasm consists of a thick, water-based fluid containing dissolved potassium salts, glucose molecules, and structural proteins. The strand travels through this fluid until it makes physical contact with a ribosome organelle. The ribosome binds physically to the messenger RNA strand. The cellular machinery reads the transcribed genetic sequence originating from chromosome 15 to manufacture a specific physical structure. The ribosome links together a chain of exactly 838 amino acids. The cell folds this long amino acid chain into a complex 12-pass transmembrane protein known to cellular biologists as the P protein. The year is 2006. The location is the National Institutes of Health in Bethesda, Maryland. Cellular biologists operate inside a biosafety level 2 laboratory. The room maintains a constant ambient temperature of 20 degrees Celsius. The ventilation system circulates air, smelling of isopropyl alcohol and sterile nitrile rubber. The researchers place cultured human melanocyte cells onto rectangular silica glass slides, measuring 25 by 75 millimeters. They utilize a laser-scanning confocal microscope to observe the physical movement of newly synthesized proteins inside the living cells. The scientists attach a green fluorescent protein, originally extracted from the Achorea victoria jellyfish, directly to the amino acid chains using covalent chemical bonds. An argon laser illuminates the sample with light waves measuring exactly 488 nanometers. The fluorescent markers reveal the complete biological life cycle of the P protein. The cellular ribosome translates the messenger RNA sequence transcribed from human chromosome 15, linking exactly 838 amino acids into a single continuous chain. This long molecular chain travels directly into the rough endoplasmic reticulum. The endoplasmic reticulum consists of a continuous network of folded phospholipid membranes. Inside this network, specialized chaperone proteins physically fold the amino acid chain into a complex three-dimensional geometrical shape. The finalized structure features 12 distinct alpha helical segments, forming a cylindrical pore. The folded protein moves into the Golgi apparatus, a cellular organelle that packages the molecules into spherical transport vesicles measuring exactly 50 nanometers in diameter. The transport vesicles carry the newly constructed P protein through the cellular cytoplasm. The vesicles arrive at the exterior membrane of a developing melanosome. The melanosome operates as a specialized storage organelle measuring 0.5 micrometers in diameter. The transport vesicle fuses with the melanosome. The 12 alpha helical segments of the P protein embed themselves permanently across the outer lipid bilayer of the melanosome membrane. The cylindrical pore structure creates a direct physical channel connecting the internal fluid of the melanosome to the surrounding cellular cytoplasm. The internal fluid of an immature melanosome contains high concentrations of positively charged hydrogen protons. This dense concentration of protons creates a highly acidic environment, registering a potential hydrogen level of 4.0. A specialized enzyme named tyrosinase resides suspended within this acidic fluid. The tyrosinase enzyme acts as the primary chemical catalyst for manufacturing brown eumelanin. To operate at maximum chemical efficiency, the tyrosinase enzyme requires a neutral potential hydrogen level of exactly 6.8. The embedded P protein channel physically opens its central pore. The protein pumps negatively charged chloride ions from the cytoplasm directly into the melanosome. The negatively charged chloride ions bind instantly with the positively charged hydrogen protons. This physical-chemical reaction raises the internal potential hydrogen level toward the neutral baseline. The neutralizing fluid activates the tyrosinase enzyme. The enzyme targets molecules of the amino acid tyrosine floating inside the organelle. The tyrosinase strips electrons from the tyrosine, converting the clear molecules into a compound called dopaquinone. The dopaquinone molecules spontaneously link together, building long, dense polymer chains of dark brown eumelanin. The pigment molecules accumulate, physically filling the interior volume of the melanosome. In a hazel eye, the specific genetic sequence inherited from the parents dictates the production of a strictly limited volume of P protein. The cellular ribosomes manufacture a low number of these 12-pass transmembrane channels. The melanocyte cell embeds this restricted number of channels into the melanosome membrane. The reduced number of P protein channels pumps a restricted volume of chloride ions into the organelle. The internal fluid remains slightly acidic. The acidic environment physically slows the processing speed of the tyrosinase enzyme. The enzyme converts a low volume of tyrosine into eumelanin before the melanosome reaches its mature structural stage. The melanosome achieves structural maturity, holding a sparse, scattered distribution of brown pigment. The melanocyte cell attaches kinesin motor proteins to the exterior membrane of the mature melanosome. The kinesin proteins consume adenosine triphosphate molecules for kinetic energy. The proteins walk physically along a network of microtubule tracts, transporting the partially filled organelle outward into the long dendritic arms of the cell. The melanocyte deposits these sparse pigment clusters into the anterior border layer of the human iris. The brown clusters sit suspended in the clear stroma fluid, separated by wide gaps of bare collagen fibers. Broad-spectrum sunlight enters the human eye at a speed of 299,792 kilometers per second. The light waves pass around the sparse brown pigment and strike the bare collagen fibers. The collagen fibers scatter the short blue wavelengths outward. The scattered blue photons physically intersect with the yellow and red wavelengths reflected by the sparse brown eumelanin clusters. This precise physical intersection of light waves creates the visual appearance of a green and gold hazel iris. Medical researchers originally identified the OCA2 gene through the clinical study of human genetic pigmentation disorders. Human patients carrying homozygous mutated sequences of the OCA2 gene produce a structurally altered amino acid chain. The cellular machinery degrades this altered chain upon detecting the structural error. The melanosome remains permanently acidic. The tyrosinase enzyme remains completely dormant. The melanocytes produce zero brown pigment. The resulting human eye develops a translucent blue color, physically exposing the red blood vessels of the retina. Medical texts classify this specific physical condition as oculocutaneous albinism type 2. The exact moderate melanin volume required for a hazel eye relies on a precise calibration of this P protein production line. To achieve this specific biological calibration, the human body utilizes a secondary genetic sequence acting as a mechanical volume dial. Genetic researchers isolate the physical mechanism of this regulatory dial in a laboratory in Copenhagen, Denmark, during the year 2008. The year is 2008. The location is the Department of Cellular and Molecular Medicine at the University of Copenhagen in Denmark. The laboratory sits inside the Panum Institute, a brutalist concrete building located on Blegdomsvej Street. The ambient temperature inside the laboratory measures exactly 21 degrees Celsius. The laboratory air smells of heated agarose powder and tris-acetate EDTA chemical buffer. Danish geneticists conduct a biological study analyzing the exact genetic inheritance of human eye color. The scientists rub sterile cotton swabs against the mucosal membrane of the inner cheeks of human volunteers. The physical friction scrapes thousands of stratified squamous epithelial cells directly onto the cotton fibers. The researchers submerge the cotton swabs into plastic microcentrifuge tubes containing a liquid lysis buffer. The chemical detergents in the buffer dissolve the phospholipid cellular membranes, releasing the complete strands of human DNA into the liquid. A laboratory technician places the plastic tubes into an aluminum centrifuge rotor. An electric motor spins the rotor at 14,000 revolutions per minute, emitting a steady mechanical hum, measuring exactly 65 decibels. The centrifugal force drives the heavy cellular debris to the bottom of the tube, leaving the purified DNA molecules suspended in the clear supernatant liquid at the top. The technicians extract this clear liquid using calibrated micropipettes. The technicians place the liquid samples into an automated thermal cycler machine. The machine utilizes thermoelectric Peltier elements to heat the liquid to exactly 95 degrees Celsius. The thermal energy physically breaks the weak hydrogen bonds connecting the complementary nucleotide base pairs, separating the DNA double helix into single strands. The machine cools the liquid to 60 degrees Celsius, allowing synthetic primer molecules to attach directly to chromosome 15. The machine heats the liquid to 72 degrees Celsius, activating the TUC polymerase enzyme. The enzyme constructs a new complementary DNA strand by attaching free-floating nucleotides to the exposed sequence. 30 automated temperature cycles produce exactly 1 billion identical copies of the targeted genetic sequence. The researchers run the amplified DNA through a sequencing machine to isolate the physical mechanism regulating the OCA2 gene. They locate the regulatory control center deep inside the neighboring HERC2 gene. The DNA sequence contains a specific region designated as intron 86. Previous genetic researchers categorized introns strictly as structural spaces separating the active, protein-coding genes along the chromosome. The Copenhagen team identifies a highly conserved regulatory sequence operating entirely within this specific spacer region. This short regulatory sequence functions as a biological enhancer. Inside the living nucleus of a human melanocyte, the long linear DNA molecule physically folds and bends into a three-dimensional loop. This physical looping brings the enhancer sequence of the HERC2 gene into direct physical contact with the promoter region of the OCA2 gene. The two distinct DNA sequences sit exactly 2 million base pairs apart on the linear chromosome, yet they touch directly in three-dimensional cellular space. Specialized proteins called transcription factors attach to this looped structure. A specific transcription factor, named HLTF, binds directly to the DNA, signaling the RNA polymerase enzyme to begin manufacturing the P protein. The researchers examine the exact nucleotide spelling of the enhancer sequence at the genetic locus designated RS12913-832. In individuals with dark brown eyes, the DNA sequence contains a thymine molecule at this exact locus on both inherited copies of chromosome 15. The atomic shape of the thymine molecule creates a tight, secure physical bond with the HLTF transcription factor. This strong chemical bond triggers the continuous, high-volume production of the P protein. The abundant P protein floods the iris tissue with dense brown eumelanin. In individuals with pure blue eyes, a cytosine molecule replaces the thymine molecule at this exact locus on both chromosomes. The cytosine molecule alters the precise geometric angle of the DNA backbone. This physical alteration repels the HLTF transcription factor. The protein detaches from the chromosome. the sequence remains permanently dormant. The cellular ribosomes produce a completely transparent anterior stroma, resulting in structural Tyndall scattering. Individuals with hazel eyes frequently inherit a heterozygous genetic combination at this exact locus. The human child inherits one copy of chromosome 15, containing the thymine sequence from one parent, and one copy containing the cytosine sequence from the other parent. The cellular machinery inside the iris melanocyte reads both sets of genetic instructions simultaneously. The single thymine allele successfully binds with the transcription factors, while the single cytosine allele repels the binding proteins. This specific physical combination reduces the overall transcription rate of the OCA2 gene by exactly 50%. The cellular ribosomes manufacture exactly half the standard volume of the P protein. The limited supply of P protein restricts the flow of chloride ions into the melanosome organelles. The tyrosinase enzymes synthesize a moderate, intermediate amount of brown eumelanin. This moderate pigment volume establishes the biological foundation for the hazel eye. The heterozygous HERC2 combination provides the necessary baseline for moderate pigment, while secondary supporting genes construct the final macroscopic geometry of the hazel iris. The human genome utilizes the TYR gene located on chromosome 11 to dictate the exact three-dimensional shape of the tyrosinase enzyme. The SLC24A4 gene, located on chromosome 14, builds specialized channels that pump calcium ions across the cell membrane. The calcium ions bind directly to the tyrosinase enzyme, physically altering its kinetic processing speed. The SLC45A2 gene, located on chromosome 5, builds sucrose transport proteins embedded in the melanosome membrane. These specific proteins alter the internal osmotic water pressure of the organelle, controlling the physical density of the eumelanin polymers. These secondary genetic sequences adjust the exact final volume and geographic placement of the pigment clusters across the 12 mm diameter of the iris stroma. These combined genetic sequences form a highly complex polygenic network, dictating the exact physical coloration of the human eye. To map the complete biological architecture of this network, researchers require a massive database of human DNA samples spanning multiple global populations. 13 years after the Copenhagen discovery, a team of geneticists working in London utilizes advanced computational algorithms to analyze the complete genomes of nearly 200,000 human individuals. The date is March 10, 2021. The location is the Strand campus of King's College London, situated directly along the north bank of the River Thames in London, England. The research laboratory maintains an air temperature of exactly 20 degrees Celsius. The ventilation system circulates air, smelling of heated silicon, and ozone, expelled by the cooling fans of running computer servers. Geneticists collaborate with researchers from the Erasmus University Medical Center in Rotterdam to execute a comprehensive genome-wide association study. The scientists analyze biological data collected from exactly 192,986 human volunteers spanning 10 distinct populations across Europe and Asia. The biological data collection begins inside regional medical clinics. Phlebotomists extract whole blood samples from the human volunteers using 22-gauge stainless steel needles. Laboratory technicians process the blood samples using centrifuge machines to isolate the white blood cells. They extract the pure DNA molecules from the cellular nuclei, Ophthalmologists capture high-resolution digital photographs of each volunteer's eyes under standardized clinical lighting. The digital cameras utilize CMOS image sensors measuring exactly 24 by 36 millimeters. The sensors contain millions of microscopic silicon photodiodes. The photodiodes convert the incoming electromagnetic light waves into exact electrical voltages. The cameras record the precise visible distribution of brown eumelanin pigment and structural tyndall scattering across the 12 millimeter diameter of every human iris. Computer engineers write a standardized mathematical algorithm to quantify this optical data. The algorithm converts the digital pixels of the eye photographs into continuous numerical scores. The scores rank the physical tissue along a precise quantitative spectrum from completely unpigmented structural blue to heavily pigmented dark brown. The geneticists load these numerical scores and the complete extracted DNA sequences into a network of supercomputers. The computer processors utilize silicon transistors measuring exactly 7 nanometers in width. The electrical current passes through these microscopic transistors billions of times per second to perform complex statistical regressions. The researchers utilize specialized microarray chips to sequence the genetic samples. The microarrays consist of square quartz glass slides measuring exactly 1.2 centimeters in width. The glass surface holds millions of microscopic DNA probes attached directly to the silica substrate. The servers process exactly 8,700,000 single nucleotide polymorphisms across the entire human genome. The software compares the specific variations in the DNA sequences with the numerical color scores of the digital photographs. The statistical analysis isolates exactly 61 distinct genetic regions directly influencing the macroscopic coloration of the human eye. Prior to this specific data computation, the global scientific community mapped a maximum of 16 regulatory genes associated with human pigmentation. The London study identifies 45 entirely new genetic sequences participating in the cellular construction of the iris. These 61 separate genes build a highly complex interconnected biochemical network inside the melanocyte cells. The computers highlight the exact biological function of these newly discovered sequences. The LYST gene, located on the long arm of human chromosome 1, constructs lysosomal trafficking regulator proteins. These specific physical proteins attach to the mature melanosomes and transport the pigment-filled organelles along the cellular microtubule tracts. The speed and mechanical efficiency of this physical transport determine the final microscopic placement of the brown pigment granules inside the stroma tissue. The data analysis isolates the DCT gene on chromosome 13. This specific sequence builds dopachrome tautomerase, a specialized enzyme operating strictly inside the acidic fluid of the melanosome. The dopachrome tautomerase physically stabilizes the intermediate chemical compounds during the synthesis of the eumelanin polymer. The precise chemical stabilization alters the final physical density of the brown pigment. The servers map the IRF4 gene on chromosome 6. This sequence creates interferon regulatory factor 4. A transcription protein that binds directly to the DNA double helix. This specific protein regulates the total biological number of melanocyte cells surviving and operating inside the anterior border layer of the human iris. In a hazel eye, the specific alleles of these 61 independent genes interact physically to construct the asymmetrical sunburst pattern. The combination of the HERC2 and OCA2 genes dictates the moderate baseline volume of eumelanin. The remaining 59 genes physically arrange this pigment across the structural collagen fibers. The regulatory proteins determine the precise geographic distance between the central brown pigment ring near the pupil and the scattered green optical wavelengths in the outer ciliary zone. The chemical interaction of these proteins controls the exact physical thickness of the radiating pigment streaks. The human genome contains over 3 billion DNA base pairs. The 61 regulatory genes sit distributed across completely different physical chromosomes. During the biological process of human reproduction, the human body generates specialized gamete cells through a cellular division process called meiosis. Inside the reproductive organs, the 46 continuous chromosomes align in physical pairs. The DNA strands break and reattach, swapping specific genetic sequences between the homologous chromosomes. This biological mechanism, known as chromosomal crossover, physically separates the 61 eye-color genes into entirely new genetic combinations. The male sperm cell carries one random assortment of these 61 genes. The female egg cell carries a completely different assortment of these genes. Upon biological fertilization, the two haploid cells fuse inside the fallopian tube to form a single diploid zygote. The newly formed genetic sequence combines the distinct parental alleles into a completely original biological network. The specific chemical interactions between these 61 separate DNA segments frequently suppress the production of the P protein in one human generation and amplify it in the next. The mechanical recombination of these distinct chromosomes establishes the precise biological pathway, allowing fully pigmented brown eyes to generate the moderate, scattered structural geometry of a hazel iris in an immediate descendant. The year is 2022. The location is the McKusick-Nathans Institute of Genetic Medicine at Johns Hopkins Hospital in the city of Baltimore, Maryland. The clinical office maintains a continuous ambient temperature of 22 degrees Celsius. The ventilation system circulates air, smelling of isopropyl alcohol and heated laser printer toner. A clinical genetic counselor sits at a wooden desk observing a liquid crystal display monitor. The genetic counselor rests their hands on a black polymer keyboard, pressing the mechanical switches to navigate a genetic database. The counselor analyzes a digital pedigree diagram mapping the exact physical inheritance of a single human family across 4 generations. The diagram utilizes specific geometric shapes to represent individual family members. Solid squares represent males with dark brown irises. White circles represent females with purely transparent structural blue irises. Shaded shapes represent individuals possessing the specific physical geometry of hazel eyes. The genetic counselor uses a plastic computer mouse to highlight a specific branch of the lineage diagram. The highlighted section connects 2 parents possessing pure blue eyes directly to a female child possessing hazel eyes. The parents carry identical homozygous cytosine alleles at the RS129138332 locus on human chromosome 15. Their cellular machinery transcribes this specific nucleotide sequence, producing purely transparent stroma tissue in both individuals. Broad-spectrum sunlight enters their eyes and hits the bare collagen fibers suspended in the stroma fluid, scattering blue electromagnetic wavelengths outward at a measurement of exactly 450 nanometers. The female child displays a completely distinct biological phenotype. The child's iris tissue contains a moderate volume of brown eumelanin pigment arranged in a central radiating sunburst pattern around the pupil. The genetic counselor inputs the complete genome sequence of all three individuals into the desktop computer. The computer processes the biological data using strict bioinformatics algorithms to isolate the 61 independent genes controlling human pigmentation. The data analysis identifies the physical manifestation of a biological process called epistasis. Epistasis occurs when one genetic sequence directly alters or modifies the physical expression of a separate genetic sequence located elsewhere in the human genome. During the biological process of meiosis, the reproductive organs of the parents generate haploid gamete cells. The 46 human chromosomes align in exact homologous pairs inside the cellular nucleus. A specialized protein structure called the synaptonemal complex physically binds the matched chromosomes together. Endonuclease enzymes physically sever the phosphodiester bonds connecting the DNA double helix at specific points called chiasmata. This mechanical severing and reattaching swaps large sections of nucleotide base pairs between the aligned chromosomes. The process, known as homologous recombination, generates entirely original genetic sequences. The male parent produces sperm cells carrying specific alleles for the 61 pigmentation genes. The female parent produces egg cells carrying a different physical arrangement of these specific alleles. The two haploid cells fuse inside the fallopian tube to form a single diploid zygote. The newly formed genome combines the stored pigment instructions from both parents. The parents pass down 59 supporting genes specifically programmed to manufacture heavy volumes of brown eumelanin. These inherited genes include the TYR sequence on chromosome 11, programmed to build the tyrosinase enzyme, and the SLC45A2 sequence on chromosome 5, programmed to build sucrose transport proteins in the cell membrane. In the physical bodies of the blue-eyed parents, the cytosine molecules at the HERC2 locus physically alter the geometric shape of the DNA strand. This altered physical angle repels the necessary transcription proteins. The disconnected proteins leave the neighboring OCA2 gene in a continuous resting state. The cellular ribosomes manufacture a purely transparent stroma. The 59 supporting genes carry their exact instructions for brown pigment directly within the DNA helix, waiting for the necessary P protein to initiate the chemical reaction. Epistasis physically masks these stored genetic instructions beneath the primary regulatory control of chromosome 15. In the newly formed zygote of the child, the homologous recombination process introduces a spontaneous single nucleotide polymorphism. A guanine molecule physically replaces a cytosine molecule near the primary regulatory sequence. This specific molecular substitution alters the folding structure of the chromatin fiber. The newly folded DNA loop creates a precise physical binding site for the HLTF transcription factor. The transcription protein attaches directly to the genetic strand and initiates the manufacturing process for the OCA2 gene. The cellular ribosomes translate the messenger RNA sequence and assemble a moderate volume of the 12-pass transmembrane P protein. The modified HERC2 sequence acts as a physical initiator for the stored epistatic genes. The P proteins embed into the phospholipid membranes of the developing melanosomes inside the iris tissue. The proteins pump negatively charged chloride ions directly into the acidic fluid of the organelles. The potential hydrogen level rises to exactly 6.8. This neutral chemical environment immediately triggers the activation of the tyrosinase enzymes built by the inherited TYR gene. The enzymes bind to copper ions and convert clear tyrosine molecules into solid brown eumelanin polymers. The melanocyte cells deposit these specific brown clusters directly into the anterior border layer of the stroma. The resulting physical structure produces a hazel eye. The genetic counselor prints a physical DNA microarray report onto a sheet of white paper using a laser printer. The printed document traces the exact biological path of the 59 supporting genes. The specific biochemical pathways for the Hazel sunburst pattern travel across 75 years of continuous human reproduction. The genetic codes survive completely intact inside the cellular nuclei of family members, exhibiting strictly pure blue or solid brown irises. The precise epistatic interference of the 61 independent genes orchestrates the sudden emergence of the moderate melanin phenotype. The physical development of this newly formed genetic combination requires active environmental stimulation to complete its structural growth. The human infant carrying this specific DNA sequence enters the world inside a brightly illuminated hospital delivery room. The melanocytes inside the stroma tissue remain in a resting state at the exact moment of human birth. The physical maturation of the hazel eye relies directly on continuous exposure to the electromagnetic radiation emitted by the ambient light sources surrounding the newborn child. The date is October 2nd, 2022. The location is a maternity ward inside Queen Charlotte's and Chelsea Hospital in London, England. The delivery room maintains a continuous air temperature of exactly 24 degrees Celsius. The mechanical ventilation system pushes filtered air smelling of iodine liquid and bleached cotton textiles. An obstetrician wearing sterile latex gloves delivers a human infant. The infant weighs exactly 3.4 kilograms and measures 51 centimeters in length. The medical staff places the infant onto a warming table heated by an overhead infrared lamp emitting thermal radiation. The infant contracts the levator palpebrae superioris muscle located in the upper eyelid. The physical contraction lifts the eyelid, exposing the clear cornea to the surrounding environment. Broad-spectrum white light from the overhead fluorescent fixtures enters the infant's eyes at a speed of 299,792 kilometers per second. The light waves pass through the cornea and enter the anterior chamber. The fluid inside the chamber consists of 99% water and 1% dissolved lactate. The light hits the front surface of the iris stroma. The melanocyte cells residing in the anterior border layer of the stroma exist in a dormant biological state at the exact moment of birth. The cellular ribosomes inside these specific cells maintain a paused production of the P protein. The melanosome organelles contain strictly clear fluid. The stroma tissue consists entirely of bare type I collagen fibers suspended in the extracellular matrix. The incoming white light waves collide physically with these suspended collagen fibers. The collision scatters the short blue electromagnetic wavelengths, measuring exactly 450 nanometers, outward into the room. The longer red and yellow wavelengths pass through the clear stroma and hit the posterior pigment epithelium. The two layers of cuboidal cells at the back of the iris contain dense black-brown eumelanin. This dark layer absorbs the long red wavelengths completely, converting the electromagnetic energy into thermal heat. The scattered blue photons exit the infant's eye. The medical staff documents the eye color as blue in the digital medical record using a plastic keyboard. The human genome requires sustained environmental stimulation to activate the 61 separate pigmentation genes inherited from the parents. The infant leaves the hospital and enters a residential home exposed to daily cycles of natural sunlight. Photons from the sun travel 93 million miles and strike the infant's retina. The photoreceptor rod and cone cells convert the electromagnetic energy into electrical action potentials. The electrical signals travel along the optic nerve to the occipital lobe at the rear of the human brain. The continuous exposure to ambient electromagnetic radiation triggers a biological response within the endocrine system. Specialized cells located in the pituitary gland release alpha-melanocyte-stimulating hormone into the human bloodstream. The hormone molecules travel through the branching capillary networks and bind directly to melanocortin-1 receptors located on the exterior membrane of the iris melanocytes. The chemical binding activates the cyclic adenosine monophosphate signaling pathway inside the melanocyte. The cellular nucleus transcribes the specific DNA sequence from locus 15q13 on chromosome 15. The ribosomes manufacture the 12-pass transmembrane P protein. The proteins embed into the melanosome membranes and pump negatively charged chloride ions into the acidic organelles. The internal potential hydrogen level rises to exactly 6.8. The tyrosinase enzymes begin stripping electrons from tyrosine molecules circulating in the cellular fluid. The chemical reaction builds long, dense polymer chains of brown eumelanin. The melanocyte cells utilize kinesin motor proteins to transport the filled melanosomes along microscopic actin filaments. The cells deposit the brown pigment clusters directly into the anterior border layer of the iris stroma. This biological manufacturing process requires exactly 36 months to reach macroscopic structural completion. During the first 12 months of life, the melanocytes deposit a moderate volume of brown pigment immediately surrounding the central pupillary margin. The physical accumulation of brown eumelanin clusters absorbs incoming visible light. The absorbed light physically mixes with the scattered blue wavelengths bouncing off the bare collagen fibers in the outer ciliary zone. The overlapping light waves produce a visible green hue measuring exactly 550 nanometers in wavelength. The physical appearance of the iris shifts from solid blue to a structural green base. During the subsequent 24 months, the melanocytes continue depositing pigment outward from the central collarette ridge. The cellular proteins transport the melanosomes along the radial collagen fibers, building dense streaks of brown eumelanin. These radial streaks form the distinct geometric sunburst pattern characteristic of the Hazel phenotype. By the 36th month, the melanocytes finalize the exact geographic distribution of the pigment. The cells establish the dense brown central ring near the pupil and the radial brown streaks across the green periphery. The human iris reaches its permanent macroscopic geometric structure. The infant's eye color fully transitions from structural blue to genetic hazel through the mechanical accumulation of specific melanin polymers. The expression of the 61 distinct genes permanently calibrates the precise ratio of scattered blue light to absorbed red light. The completed biological structure maintains this specific pigment density for the remainder of the human lifespan. The growing child navigates through different physical environments containing varying levels of ambient illumination. The child walks from a brightly lit outdoor playground directly into a darkened residential hallway. The sudden drop in ambient photons triggers a reflex within the parasympathetic nervous system. The nervous system sends electrical impulses to the radial smooth muscle fibers located inside the iris stroma. The muscle fibers physically contract, pulling the inner tissue outward to widen the central pupil and capture more incoming light. The physical expansion of the central pupil mechanically compresses the outer stroma tissue, pushing the suspended brown pigment clusters closer together and dynamically altering the visible structural color of the hazel eye. The year is 2023. The location is an optics research laboratory inside the Herbert Wertheim School of Optometry and Vision Science at the University of California, Berkeley. The laboratory maintains an ambient air temperature of exactly 20 degrees Celsius. The ventilation system circulates air smelling of isopropyl alcohol and heated electrical insulation. An optical researcher sits across from a human volunteer possessing the 61 distinct genetic markers for hazel eyes. The volunteer places their mandible into the plastic chin rest of a digital pupillometer. The digital pupillometer contains an infrared light-emitting diode emitting electromagnetic wavelengths measuring exactly 850 nanometers. The device uses a charge-coupled device camera to record the exact physical diameter of the human pupil at a rate of 60 frames per second. The researcher activates a xenon arc lamp mounted directly to the heavy steel examination table. The lamp generates a broad-spectrum white light measuring exactly 5,000 lumens in luminous flux. The photons travel across the examination table and enter the clear cornea of the volunteer. The light passes through the aqueous humor, moves through the central pupil opening, and strikes the photoreceptor cells in the retina. The photoreceptor cells convert the photon impacts into electrical action potentials. The electrical signals travel along the optic nerve directly to the Edinger-Westphal nucleus, located in the human midbrain. The midbrain processes the high-intensity light signal and activates the parasympathetic nervous system. Parasympathetic nerve fibers travel back to the human eye and release a neurotransmitter chemical called acetylcholine. The acetylcholine molecules bind to muscarinic receptors located on the sphincter pupillae muscle inside the iris stroma. The sphincter pupillae consists of circular smooth muscle fibers arranged directly around the central pupillary margin. The chemical binding causes these circular muscle fibers to physically contract. The contraction pulls the inner edge of the iris inward, reducing the pupil diameter to exactly 2 millimeters. This mechanical reduction of the pupil physically stretches the iris stroma across the anterior chamber of the eye. The stretching expands the surface area of the 12-millimeter iris tissue. The physical expansion pulls the microscopic clusters of brown eumelanin pigment further apart from each other. The physical distance between the individual melanosomes increases significantly. The physical stretching pulls the deep radial furrows of the iris completely flat against the clear fluid of the stroma. The increased physical distance between the pigment clusters exposes a massive volume of bare type 1 collagen fibers to the incoming white light. The photons hit the exposed collagen strands, triggering intense tyndall scattering across the entire surface area of the iris. The collagen scatters short blue wavelengths, measuring 450 nanometers, outward toward the researcher. These scattered blue photons physically intersect with the yellow light waves, reflecting off the widely spaced brown pigment clusters. The overlapping electromagnetic waves produce a highly visible green hue, measuring exactly 550 nanometers. The mechanical stretching of the tissue physically maximizes the scattered structural green color of the hazel eye. The researcher turns a rotary potentiometer dial connected to the xenon arc lamp power supply. The dial increases the electrical resistance in the copper wiring, dropping the light output from 5,000 lumens down to 50 lumens. the ambient illumination inside the laboratory plummets. The photoreceptor cells in the volunteer's retina detect the immediate drop in incoming photons. The biological signal travels to the hypothalamus, activating the sympathetic nervous system. Sympathetic nerve fibers travel through the superior cervical ganglion and release the neurotransmitter norepinephrine. The norepinephrine molecules bind directly to adrenergic receptors located on the dilator pupillae muscle. The dilator pupillae consists of radial smooth muscle fibers extending outward from the collarette ridge to the outer ciliary edge of the iris. The chemical binding forces these radial muscle fibers to rapidly contract. The physical contraction pulls the inner pupillary margin outward, retracting the iris tissue away from the center of the eye. The central pupil diameter expands to exactly 8 millimeters to capture the remaining ambient light. The physical expansion of the central pupil forces the surrounding iris tissue to compress. The entire volume of the 12-millimeter stroma physically compresses into a tight circular band measuring exactly 2 millimeters in width near the outer edge of the cornea. The physical compression forces the scattered brown eumelanin clusters to pack tightly together. The microscopic distance between the pigment granules decreases until the melanosomes physically overlap. The dense packing of the brown pigment completely covers the underlying collagen fibers. The incoming light hits the dense brown pigment ring. The tight physical arrangement of the eumelanin polymers absorbs the incoming visible wavelengths completely. The biological tissue converts the electromagnetic energy directly into microscopic amounts of thermal heat. The physical covering of the collagen fibers halts the Tyndall scattering process. The blue wavelengths cease reflecting outward. The optical researcher observes the physical color of the volunteer's eye shift from structural green to solid dark brown in exactly 2.5 seconds. This mechanical tissue compression and expansion happens continuously throughout the daily life of the human subject. A person walking from a brightly sunlit pavement into a shadowed residential hallway triggers this exact biological mechanism. The fluctuating ambient photons dictate the exact diameter of the central pupil. The pupil diameter dictates the exact physical density of the eumelanin clusters inside the stroma. The variable pigment density dynamically shifts the ratio of absorbed red light to scattered blue light. The perceived color of the hazel eye operates strictly as a mechanical optical effect driven by the involuntary contractions of smooth muscle tissue. The physical structure of the iris remains mechanically altered until the ambient lighting conditions change again. The human visual cortex processes these dynamic structural changes through complex neurological pathways. The biological observer simultaneously processes the exact wavelengths of light bouncing off the physical objects surrounding the human subject. Humans manufacture textiles dyed with specific chemical pigments to wear as clothing. The reflected electromagnetic wavelengths from these dyed fabrics enter the observer's eye alongside the scattered wavelengths originating from the hazel iris. The human brain processes both sets of intersecting electromagnetic waves at the exact same time inside the occipital lobe. The year is 1839. The location is the Gobelins Manufactory on the Avenue des Gobelins in Paris, France. The air inside the textile workshop smells of boiling water and wet sheep wool. French chemist Michel-Eugène Chevreul stands beside a large wooden loom. He directs a team of workers weaving large tapestries. The workers weave strands of spun wool, dyed with natural pigments. They place a yellow wool thread, dyed with weld plant extract, directly next to a blue wool thread, dyed with woad leaves. Chevreul observes the reflected light waves bouncing off the adjacent threads. He records that the physical proximity of the specific textile colors alters the human perception of the individual hues. He publishes a scientific text titled The Law of Simultaneous Color Contrast. We apply Chevreul's 19th century optical law directly to human biological interactions today. Two humans stand exactly 1 meter apart inside a room, illuminated by a tungsten halogen incandescent bulb. The bulb operates at a color temperature of 3200 Kelvin, emitting a continuous beam of broad-spectrum white electromagnetic radiation. One human possesses hazel eyes, featuring the specific moderate distribution of brown eumelanin and clear collagen fibers across the iris stroma. This individual wears a cotton textile shirt dyed with copper phthalocyanine. The synthetic chemical dye absorbs the red and blue electromagnetic wavelengths from the tungsten bulb. The dyed cotton fibers reflect green light waves measuring exactly 530 nanometers in length. The tungsten light simultaneously hits the hazel irises of the individual. The physical collagen fibers inside the iris stroma scatter blue wavelengths measuring exactly 450 nanometers. The moderate clusters of brown eumelanin reflect yellow wavelengths measuring 570 nanometers. The green wavelengths from the dyed cotton shirt and the scattered wavelengths from the hazel irises travel across the 1 meter distance simultaneously. The three distinct sets of electromagnetic wavelengths enter the open pupil of the observing human. The photons travel through the liquid vitreous humor and strike the observer's retina. The retina contains 6 million specialized cone cells concentrated in the central macula. The cone cells contain complex biological proteins called photopsins. The incoming photons strike the photopsin proteins, triggering an immediate physical change in the molecular shape of the protein. This physical change forces the cone cells to release glutamate neurotransmitters. The neurotransmitters generate electrical action potentials inside the adjacent bipolar cells and retinal ganglion cells. The retinal ganglion cells utilize a biological processing mechanism called lateral inhibition. When the 530 nanometer green wavelengths from the cotton shirt hit a specific cluster of cone cells, the resulting electrical signal actively suppresses the electrical output of the immediately adjacent cone cells processing the edge of the shirt. This localized neurological suppression heightens the physical contrast at the exact visual boundary between the green fabric and the human face. The electrical signals travel from the ganglion cells into the optic nerve. The signals travel along the myelinated axons of the optic nerve at a speed of exactly 100 meters per second. The electrical impulses enter the lateral geniculate nucleus located inside the human thalamus. The thalamus routes the electrical data to the primary visual cortex, classified by neurologists as the V1 area, located in the occipital lobe at the rear of the human skull. The V1 area contains specialized neurons designated to process specific spatial frequencies and color boundaries. The processed electrical signals proceed directly to the adjacent V4 area of the visual cortex. The V4 neurons process the overlapping signals originating from the green shirt and the hazel eyes. The neurological processing of the large green visual field generated by the copper phthalocyanine dye physically alters the observer's perception of the smaller visual field containing the hazel eye. The visual cortex actively highlights the scattered blue and yellow wavelengths originating from the stroma to maximize the visual contrast against the green fabric. The human observer consciously perceives the hazel eyes as displaying a vibrant, solid green hue. The hazel-eyed individual removes the green cotton shirt and puts on a shirt dyed with red iron oxide. The iron oxide pigment absorbs the blue and green wavelengths from the tungsten bulb. The dyed fabric reflects long red and orange wavelengths, measuring exactly 650 nanometers. The tungsten light hits the brown shirt and reflects these long wavelengths toward the observer. The long red and orange wavelengths enter the observer's retina. The cone cells generate a completely different pattern of electrical action potentials. The lateral inhibition in the retinal ganglion cells shifts its suppression boundaries. The V4 area of the visual cortex receives the newly generated electrical data. The dominant red and orange wavelengths from the fabric prompt the visual cortex to highlight the identical long wavelengths reflecting off the brown eumelanin clusters near the pupillary margin of the hazel eye. The observer consciously perceives the exact same hazel eyes as displaying a solid, dark brown hue. During this entire optical exchange, the physical structure of the hazel eye maintains its exact cellular geometry. The specialized melanocytes in the anterior border layer maintain their exact resting baseline volume of eumelanin. The collagen fibers inside the stroma maintain their exact physical thickness of 20 nanometers. The central pupil maintains its exact mechanical diameter. The shifting color exists entirely as a neurological calculation generated inside the occipital lobe of the observer. The reliance on the observer's visual cortex makes human eyewitness testimony regarding hazel eyes biologically inconsistent. Police detectives record conflicting physical descriptions from multiple witnesses observing the exact same individual under different ambient lighting and clothing conditions. One witness writes a sworn statement describing a suspect with green eyes, while a second witness writes a sworn statement describing the identical suspect with brown eyes. Investigators bypass this neurological variability by extracting pure genetic material left at crime scenes. They transport the cellular DNA to specialized forensic laboratories to determine the suspect's physical eye color through direct biochemical sequencing algorithms. The year is 2010. The location is the Department of Forensic Molecular Biology at the Erasmus University Medical Center in Rotterdam, Netherlands. The laboratory maintains a constant air temperature of 21 degrees Celsius. The ventilation system circulates air, smelling of sodium hypochlorite bleach and powdered latex gloves. Forensic geneticists Manfred Kaiser and Susan Walsh stand beside a stainless steel laboratory workbench. They process biological evidence collected from physical crime scenes. They utilize sterile steel forceps to handle a piece of cotton fabric stained with human blood. The dried red blood cells and white blood cells adhere directly to the woven cotton fibers. The geneticists cut a 1 centimeter square from the fabric using sterilized steel scissors. They place the fabric square into a plastic microcentrifuge tube holding exactly 2 milliliters of fluid. They add a chemical solution containing proteinase K, a specialized digestive enzyme. They place the tube into a heated incubation block operating at 56 degrees Celsius. The thermal energy accelerates the enzymatic reaction. The proteinase K molecules physically break the peptide bonds holding the cellular membranes together. The destruction of the cellular walls releases the intact human DNA strands directly into the surrounding liquid solution. The technicians isolate the pure DNA using a centrifuge spinning at 14,000 revolutions per minute. They extract the genetic material to test a newly developed forensic phenotyping tool named IrisPlex. The Irisplex system consists of a customized biochemical assay and a digital computer algorithm designed to predict a suspect's physical eye color strictly from biological evidence. The human genome contains over 10 million single nucleotide polymorphisms, designated scientifically as SNPs. The geneticists program the Irisplex assay to target exactly 6 specific SNP loci known to regulate human melanin production. The targeted genetic markers include the RS129133832 locus on the HERC2 gene, the RS1800407 locus on the OCA2 gene, and the RS128963999 locus on the SLC24A4 gene. The assay also isolates specific regulatory locations on the SLC45A2, TYR, and IRF4 genes. The technicians load the purified DNA sample into a thermal cycler machine. The machine executes a PCR amplification protocol, heating and cooling the liquid to build millions of identical copies of these 6 exact genetic targets. A capillary electrophoresis machine sequences the amplified DNA fragments. An argon laser illuminates fluorescent chemical tags attached to the nucleotides. A charge-coupled device camera records the specific color of the emitted light photons, identifying the exact presence of adenine, thymine, cytosine, or guanine at the 6 targeted locations. The digital camera transmits this precise nucleotide data through copper cables into a desktop computer workstation. The computer runs a specialized software program containing a multinomial logistic regression algorithm. The mathematical algorithm calculates the statistical probability of a physical phenotype based strictly on the specific combination of those 6 DNA markers. The software generates a digital report classifying the biological suspect into one of 3 absolute physical categories. The system outputs a prediction of pure blue eyes, pure brown eyes, or an intermediate eye color. When the machine processes genetic material originating from suspects with solid brown or solid blue irises, the mathematical algorithm achieves a prediction accuracy rate of exactly 93%. The solid colors rely on straightforward genetic instructions dictating either a massive volume of the P protein or a complete absence of the P protein. Law enforcement agencies across Europe and the United States adopt the Irisplex system to generate physical suspect profiles. Police detectives print the digital phenotype reports onto standard paper and distribute the predicted physical descriptions to patrol officers and local media outlets. The forensic prediction system encounters severe mathematical disruption when processing biological material deposited by suspects possessing hazel eyes. The machine sequences the DNA of a hazel-eyed individual and detects a highly complex, heterozygous combination of alleles across the 6 targeted loci. The computer reads a thymine molecule paired directly with a cytosine molecule at the primary HERC2 regulatory switch. The sequencing reveals a mixture of active and dormant alleles across the 5 supporting genes. The computer inputs these conflicting biological variables into the multinomial logistic regression model. The mathematical formula distributes the statistical probability evenly across multiple physical outcomes. The software outputs a prediction of intermediate eye color, accompanied by a confidence interval dropping to 50% or lower. The specific polygenic balance required for moderate melanin completely disrupts the exact digital categorization parameters of the software. The resulting phenotype report provides inadequate physical details for criminal investigators relying on definitive anatomical descriptions. The mathematical failure stems from the limited scope of the irisplex targeting parameters. The algorithm calculates the statistical output based exclusively on 6 genetic markers. The biological reality of a hazel eye requires the precise epistatic interaction of exactly 61 distinct genes operating simultaneously. The remaining 55 genes dictate the exact physical distribution of the eumelanin clusters across the structural collagen fibers. The forensic software operates entirely without the genetic data controlling the central sunburst pattern, the dense collarette ring, or the peripheral clear zones of the stroma. The physical appearance of the hazel eye relies on the dynamic mechanical contraction of the smooth muscle fibers surrounding the pupil. The ambient lighting at a crime scene forces the sphincter pupillae muscle to contract or relax, physically stretching the tissue and altering the density of the suspended eumelanin granules. The genetic algorithm processes only the base biological instructions for melanin volume. The software possesses zero capacity to calculate the physical stretching of the tissue or the environmental lighting conditions altering the Tyndall scattering process at the time of an eyewitness observation. Hazel eyes remain a persistent forensic limitation for the most advanced criminal genetic software. The intermediate phenotype consistently defies the digital prediction algorithms utilized by global law enforcement laboratories. To track the precise physical existence of this specific biological trait, scientific researchers bypass these predictive algorithms entirely. They shift their focus from individualized forensic software directly to massive demographic census data, mapping the physical traits of human populations across global continents. The year is 2024. The location is the United States Census Bureau headquarters, located in Suitland, Maryland. The office building maintains an internal air temperature of exactly 21 degrees Celsius. The mechanical ventilation system circulates air, smelling of floor wax, and the ozone emitted by running computer servers. Demographers sit at workstations utilizing liquid crystal display monitors. They compile human phenotype data collected from national and international medical surveys. The statisticians input the biological data of exactly 8 billion humans currently inhabiting the Earth into a central database. The statistical software calculates the physical distribution of structural eye colors across the global population. The digital datasets reveal the exact macroscopic geometry of human irises worldwide. Exactly 79% of the global human population possesses solid brown eyes. These individuals carry the functional genetic sequences required to manufacture dense volumes of brown eumelanin pigment inside the stroma tissue. Approximately 10% of the human population possesses pure blue eyes. These individuals carry the mutated HERC2 sequence, resulting in completely transparent stroma tissue containing bare collagen fibers. The software processes the remaining percentages to identify intermediate eye colors. Exactly 5% of the global population possesses the specific moderate melanin distribution required to form hazel eyes. The demographers map this specific 5% onto a digital projection of the Earth. The data points concentrate heavily along the geographic borders of the Mediterranean Sea. High frequencies of hazel irises appear in human populations residing in modern-day Spain, Italy, and Greece. The genetic markers track across the coastal cities of North Africa, appearing continuously in populations living in Morocco and Algeria. The trait distributes throughout the Levant region, specifically within the borders of Lebanon and Syria. These exact geographic concentrations align physically with the ancient maritime trade routes established during the Bronze Age. Humans carrying the mutated genetic sequences from the Black Sea physically walked and sailed into territories populated by humans carrying the ancient genes for heavy melanin production. The continuous physical reproduction between these distinct populations mixed the 61 regulatory genes into the shared DNA pool. The continuous genetic recombination established the polygenic network required for the Hazel phenotype. The demographic data tracks these exact genetic sequences across the Atlantic Ocean. European migrants sailed wooden ships across the ocean beginning in the 15th century, bringing the heterozygous chromosomal combinations to the Americas. Today, the USA reports a hazel eye prevalence of exactly 18% within its national borders. In South America, the national census data of Brazil records high frequencies of hazel eyes in the southern states of Paraná and Rio Grande do Sul. Human migrants from Portugal, Italy, and Lebanon settled in these specific regions during the 19th and 20th centuries, depositing their specific genetic alleles into the regional population. Inside biological laboratories, scientists study these physical traits at the molecular level. The year is 2025. The location is the Broad Institute in Cambridge, Massachusetts. Cellular biologists utilize clustered, regularly interspaced, short palindromic repeats, a gene editing technology known as CRISPR. They extract human melanocyte cells using stainless steel needles and place them into glass petri dishes. The scientists program a guide RNA sequence to target the specific rs129138332 locus on human chromosome 15. They inject the guide sequence and a Cas9 nuclease enzyme directly into the cellular nucleus using a glass microscopic pipette. The Cas9 enzyme physically cuts the DNA double helix at the targeted location. The scientists introduce a homology-directed repair template into the nucleus. The cellular repair mechanism substitutes a cytosine molecule with a thymine molecule. The cellular ribosomes immediately begin manufacturing the P protein. The melanosomes inside the cell fill with dense brown eumelanin. The scientists successfully alter the primary regulatory switch controlling melanin production. The available biochemical tools limit the scientists to editing one specific genetic locus per procedure. The physical distribution of the brown pigment across the 12 mm human stroma requires the simultaneous coordination of exactly 61 distinct genetic sequences. The 61 genes sit on completely different chromosomes. The scientists halt the experiment after modifying the primary switch. The specific radial anatomical pattern of the hazel eye develops strictly through the mechanical process of natural human meiosis and chromosomal crossover. The physical construction of the hazel eye relies entirely on this continuous biological chain of human reproduction. Two humans undergo sexual reproduction. The 46 human chromosomes swap genetic sequences through homologous recombination. The female parent gestates the diploid zygote inside the uterus for exactly 40 weeks. The infant enters the physical environment. Ambient electromagnetic radiation activates the endocrine system. The melanocyte cells deposit the moderate clusters of brown eumelanin into the anterior border layer of the iris stroma. A photon of light travels 93 million miles from the sun at a speed of 299,792 kilometers per second. The photon strikes the clear cornea of the human eye. The light wave passes through the aqueous humor and enters the stroma tissue. The short blue wavelengths, measuring exactly 450 nanometers, strike the suspended collagen fibers. The physical collision scatters the blue photons outward. The scattered blue light physically intersects with the yellow and red wavelengths, reflecting off the adjacent clusters of brown eumelanin. The overlapping electromagnetic waves produce a visual frequency measuring exactly 550 nanometers. The combined light waves travel through the air and enter the eye of a human observer. The photoreceptor cone cells in the observer's retina convert the photons into electrical action potentials. The electrical signals travel along the optic nerve into the occipital lobe. The human visual cortex processes the electrical data and registers the physical presence of a rare, structural green and gold hazel iris.